Thank you.
These are chicken stays there that be big.
Anna?
Do you need any other?
Yeah. We're gonna have 2 folders on the desktop for presentations, task force AM, for task AM, task PM. Okay, and then also, I don't know who's gonna be doing help with opening people's box that are up. Okay. And you want me to? I don't know who's doing what. I'm here, and,
I'm doing intros in the afternoon,
okay.
which might mean I'm gonna be. I don't know how does it work that it can be online, but whether you open up to you to everybody.
Let me show you how that's gonna work.
Gotcha.
Here. So you have to share screen 2, which is that screen. Right?
The big guy.
The big guy,
Okay.
screen 2. Okay, that screen is gonna have our. Right, well, yeah, okay. And there are 3 screens that we see, and then there's that.
Right.
So when you hit the share screen, you're gonna hit the screen.
That'll be for the presentation.
Correct. And then when people start the presentations, with my. I did. Hearing. That's the screen. And then. Is the one we're gonna move over to. There. Okay. And that's gonna have a whole bunch of people. Somebody. All right. And then if people want to be able to use a pointer, they have to come back. Yes. It does. Not only show-
Yeah, it doesn't work for all of them. Well,
This is the only one that works for one. Okay.
yeah. Should be here. Where are we keeping presentation?
Okay, so I made it. Nothing for you, John. Hello?
Hey, how are you doing?
Good. I just, sorry, I haven't. We have a million screens. Even there.
They're still there. Because you weren't. Sure.
The screen to share is screen 2. I'm having a screen. So when you hit share. And then you open it. How do you do something? How to get better than I used
Yeah.
Where's the audio gonna be?
Or something.
See, there's
Yeah.
microphones all over.
Oh, yeah, no, you're just standing up here.
Right, and if people use this. And I can do it again. Right. Oh, sweet, okay. I have to give this pointer. Okay, yeah. Only the people that can improve it.
This one's the diabetic way to start.
So.
Three different
So
stuff
I
like
guess.
this, and we also have that.
All right, cool. So you want to just put like, maybe put some more sugars on top of these and more taste on that, or do you want to just have it like this?
Yes.
Just put a couple maybe on the side.
Yeah, it's not.
It seems like that, 'cause it's in-
Well, help yourself.
Right. Thank you. Thank you.
Yeah.
Oh, Evan, you found my. And then when people come.
Did Todd bring it? Todd got it. I thought he might have brought his area.
Oh, I don't know. Oh, he did. What else was possible? Gotcha. Thank you.
Small.
Oh, thank you. No, I feel like more like. Of course. Wow.
Damn, how much is this?
Sometimes
I
it's.
feel like I did.
You guys have the poison one. Yep. Oh, one thing we got. The poison. That's that part.
Yeah.
Oh, but you can look it out. Yeah. That's great.
You happen to have a penny?
Sure did.
I'll borrow it.
Oh, here, we have to open these things. I was.
Sabrina, we're not gonna go get more ice.
Okay, is there an ice? There might be an ice machine in there. Oh.
Alright, oh, that's fine.
Oh, my God. Do you have a knife on them?
You were setting up, I walked right in.
And I gave you that swing down.
Yeah.
Need the other side. Make the top both sides here. Thank you.
Yep.
Okay.
Oh, yes.
Jump on. Well, for the top falling off.
Yeah.
All right, guys. I'll get something.
I got it.
Hello.
Got their own branches.
Yep.
There you go.
Thank you.
Yeah.
We got cinnamon and blueberry.
How's your drive down? Oh, not bad. It was kind of after traffic yesterday. We got into the area at like 9:30.
Okay.
Crash. Here we are.
Where'd you stay?
You're off the bridge.
Okay.
Yeah.
Yeah.
Yeah, for some reason, it was kind of I surprised myself. For the morning, it was just turning at 8, be here at 7:30. Oh. And Charlie.
Yeah.
Looked at it last night. Dude, why do you think you were alone for 5? I got an extra hour of sleep. That's good. That's good.
It's kind of that.
Yeah, yeah.
We all like pastries. It's good.
Yeah.
Oh, we got some fruit here.
Yeah.
Hey, Margaret.
I know. Maybe Dr. Jones.
Oh, really?
Yeah.
Good, good.
Yeah. Yeah, it's great. Yeah.
Oh.
Thank you, I'm gonna see you talking about it. Good.
I think we're getting there here. And then I'm trying to tell you these things out. We get them condensed, I think we'll be better.
Yes, they're using that. See it. There's the-
Let me get it.
Yeah.
No, it's out a little bit.
Yeah.
But thanks. Good. You get, yep. Was it last week at Patrick's or?
Yeah.
Yeah.
It was good.
Good,
Yeah,
good.
yeah, he made it.
That's awesome.
He wasn't gonna.
Okay.
Yes. but-
Nice.
And we were, he was observed.
Okay.
He likes to be observed.
Okay, that's it.
Yeah. it's like-
Yeah, no, and we locked down. Maybe
I know.
it's nice.
We leave now.
Yeah.
We lost.
That's great, last year. Yeah. Of course, like, I'm just never.
Yeah.
But I think it was, what was vertical because of the snow or something, like there might have been.
Was some, yeah.
Vince couldn't get here.
Yeah,
Yeah.
so.
Yeah.
It's improved.
You know, do it just. Albany.
Yeah. You want to be. You want to. Flash drive again?
Yes. That's dangerous. No.
Yeah.
Could we, here's a flash, I didn't do that. What I mean, chance never.
Yeah, oh, yeah. I'll go through this, you know, as soon as we're done with this.
All right.
Yeah. Good.
Please go eat. Hey, everybody.
Yeah.
I'm myself. How are you?
Is that?
He doesn't, I don't really care, if he does, he doesn't, but like, this, he just.
The only thing I would say. Just, you know, that is, I don't want to get the real. Like, we can shot, I mean. Feel like he's gonna down. I'm afraid he'll kind of.
Yeah, I think it's. I'm sure it's- but I think the kids, but I'm followed up.
How are your?
Yeah. It's like, oh yeah, it's supposed to be.
Thank you. And basically, like she didn't.
That makes sense.
You know, it's like, say, it's like, whatever. Yeah. Yeah, no, it's not.
Many, within the line, many, they had it like this. In terms of proposal, I think initially. On that, the very, it's putting a high. There's been that so much time.
Yes.
I've enjoyed the work. She had done like, and she was really.
Yeah.
And realistic. I mean. Well,
Yeah, I don't know if I asked yesterday, you know, years I did. Yeah, which, I it's just frustrated, you know, didn't do her, so he was down here. Hey, sure.
Yeah, well, they do.
I know. I'm sick, just so my heart is. All right.
No.
No. Yes. We have those.
Oh, gosh. Yeah. Figuring out.
Change. So when I get it, say,
Which is. Not even.
My seats for lunch.
And she was not deal with it.
Yeah. Scooching to take now it's just like this is my yeah. So I got that turn on or whatever. Yeah, it's something else. Like yeah, I turn it. I know.
Yeah. Because that long, you have to turn around.
Yeah. You make You may have different. Related to all of it. Yeah, but I mean,
Yeah. Yeah, it's pretty nice.
Or you can take no. Yeah, no.
Yeah.
Talk about the next one. The volcano, which are volcano. Yeah.
I guess it's better she'll have a relationship.
Sharon. Wait.
I stayed in the last one there.
Yeah, it's like kind of good.
You know, it's. CBH. Last day. Yeah.
That's not.
Yeah. Yeah.
For that, yeah.
So like kind of two things. I'm glad that we were like, they're probably over each other.
Yeah. Well.
Yeah.
Lives only how do that taste?
Yeah. Yeah.
All this stuff should be advisory because I don't know how to. It's ready to be like.
I think it was.
That's good. Always not saying everything's like. Yeah. You know, yeah. They stay. Numbers. It's like a.
Yeah, but it's like. Doing something where.
Yeah, that was so.
That's so nice. That moment. Nice to meet you. I said, "Oh, she yes. Now, on Saturday, it's just like she was saying like, she was all kind of like dark out who, it's like then they gave this. Yeah, literally, like, what's that? She's like, New Jersey. No. So she'll play, she plays tonight, tomorrow, so 5:30 to 9:30 and 8:80. Yeah, but now, yeah, but you can't. Yeah, but because things are a little different, it's one each day. Yeah. And you don't have first time. That place. Two teams.
There's no way to manage.
Oh.
The
two teams. And then there's going to be.
Good morning. Welcome to VIMS and Virginia Waterborne Women's Task Force meeting 2023 version, recap of 2022. I'm Kim Reece. I'm a professor here at VIMS and chair of Aquatic Health Sciences. I'm glad to welcome those of you in person and those of you on Zoom. Hopefully those of you on Zoom can hear us okay. There are microphones throughout the room, so please let us know if you cannot hear us. If anybody on Zoom can kind of give a thumbs up or let me know. Can you hear us?
There
Great.
we go.
Yay, thank you. Okay, throughout the room, there are mics, so hopefully speak clearly and loudly when you ask questions in the room. Those of you on Zoom, my graduate student, Savannah Mapes, will be monitoring the questions on Zoom. This morning, Jackie Friedman will be introducing speakers and being our moderator in the morning and keeping us on time. You'll have to bring me in. Thank you for joining us. In the afternoon, Evan Jurgen will be our moderator, and Evan will keep us all on time in the afternoon. Moderate and introduce the speakers in the afternoon. A few logistics for those of you here in the room. Restrooms, important thing. Out the door to your right and on the left are the restrooms right around the main VIMS desk. In the back of the room, hopefully you found the caffeine. Savannah was nice enough to go back over and get us a teapot. I ordered hot water, but it didn't come, so the teapot from my office is there with some hot water and if you there for some tea, and I brought some tea bags from my office. There's coffee, water, pastries, and some protein bars to keep you going through the morning. Please help yourself and feel free to get up and get more caffeine if you need it. The name tags that you have, please return them at the end of the day. Please try to remember, we have to pay for them if you don't. It's not the end of the world, but please try to remember to return them at the end of the day. But it helps us know each other and know who's from where and what you're doing. Margaret, Todd, can you think of anything else for the logistics for right now?
Where's the camera? Are we being filmed as we're speaking?
It started the
Okay.
recording as soon as I started
Okay.
the Zoom this morning.
Gotcha.
There's also a lot of other little recordings from when people tried out Zoom. So
Okay.
we have lots of recordings of the Virginia Carnival Task Force 2023 meeting. Anyway, so now I'd like to introduce Danielle Schools. She is going to give us a little introduction and background of the Shellfish Center. Shellfish safety.
Yeah, so good morning, everybody. Good morning, computer world folks. My name is Danielle Schools. I'm the director for Division of Shellfish Safety and Waterborne Hazards. Our program, obviously, we dabble in marine and freshwater issues as far as HABs, and all of you, I'm sure, familiar with Todd and Margaret, and also Amy, who supports Margaret and Todd in their efforts, as well as Evan and Jackie. So I'm gonna keep it short because I know we've got a packed agenda and we need as much time as we can have. I'm really excited about this. It's great. I still feel like we're rebounding from COVID. Every time I get in a room with a bunch of people, I'm like, yay, maybe we can finally get past this thing. Our focus is really on the public health aspect of these occurrences. And one of the things I think we learned with COVID was demand is not always met with the resources. So I just wanted to just take a moment to just kind of acknowledge the amount of work that everyone has done in participating in these activities, from identification of the blooms to sampling to response, education, to the laboratory support. Each piece has a critical element as far as trying to protect the public and promote awareness of these issues. I think it's something that, climate obviously is changing. Look at what we were at yesterday. And some of the guys who focus more on shellfish called yesterday morning and were like, the water temperature is above 50. And I'm like, ooh, it's February. So, the concern is there. It's only going to increase. I just, again, want to applaud you all's efforts because you've done an enormous amount of work with very limited resources. I think a lot of times when you're public health game, there's very few thank you. There's more the other end where there's demand. What are you doing? Why are you not doing this? I want this done now. And I can understand that. I'm someone who enjoys the waterways, I like the kayak, I like the tube, I like the fish, I like to eat shellfish. The risk is real. And we all know that. And so I think, the success of this program today is because of you folks and those who are joining us from computer world. So I just wanted to take opportunity to just express my gratitude on the efforts that you all have made in pursuing this task and keeping folks safe. So I'll just kind of wrap it up with that. Excited to hear the speakers. And, I said something to some of the students that Leah was introducing me to. Please, at any time, interact with us. I know you all interact with Todd and Margaret and Evan and Jackie that's new to the marine science program. But if any of us in our group can be a resource to you for whatever reason, please don't ever hesitate to reach out and chat with us, because you have an opportunity to educate us as well. It really is a learning environment. We're kind of learning as we go. There's been a lot of teaching moments. I know when we responded to the mats up on the Shenandoah River, I learned a lot. I think now I know all of the bends and crooks in that river that I never knew before. because that experience, because Margaret and I think we've worked on that response throughout the whole weekend. So it's a heavy task and it's a heavy load. One of the things that I have said with my shellfish folks on the side of the house is we do a ton of work with very limited resources and we're only successful because of the people. And I think with the response to the HABs and the work that's being done is only because of you all. So I just can't thank you enough for the work that you do. So I guess let's kick it off and excited to hear the speakers.
Okay, first up this morning, we'll have a session from Juliette Smith's lab here at VIMS for her marine lightning talk, as a recap of what her lab has been up to. Up first, we'll have Joshua Garber, who is a graduate student in Juliette's lab, and his talk is the optimization of SPATT production methods and determination of storage viability.
Everyone, my name is Todd. I am a master of biotoxin and Juliette's lab here at VIMS, and today I'll be talking about two sets of experiments that I ran over the course of last year. And those are the optimization of the SPATT production method and determination of storage viability. So the first one, the production method, the objective of this set of experiments was to find the production of SPATs, which are solid phase absorption toxin trackers that we use in our lab. Previously, the method that we used took 30 days to make a batch of SPATs. This involved soaking the resin in methanol for 24 hours, followed by water for 24 hours. With the new method, we can make them within about 2 hours. I've shortened the resin soaking segments down to 15 minutes of methanol, followed by 15 minutes of water. And so to test if these are just as effective, for the procedure, I made a total of 18 SPATs, 9 of them using the longer method and 9 of them using the shorter method. Triplicates of each type were left out for either 1 day, 7 days, or 14 days, and I attached them all onto this PVC quadrant and then deployed them off the pier into the York River. And I did this set of experiments twice, once in January and once in September, just seeing if there were different toxins out there at different times. And these are the results that I found. So on the left is the results from January, and on the right is the results from September. And overall, the finding was that the SPATs made with the short method were just as effective, if not more effective, than the SPATs made with the long method. Any of the groups of 2 bar graphs that have an asterisk show a statistically significant result difference between the short and the long method. So on the left graph from January, I found Pectenotoxin 2, which is shown by graph A, oxalic acid in graph B, and Phytoplankton 1, which is shown in graph C. In September, I found essentially the same toxins. I found Pectenotoxin 2 and oxalic acid, but this time there was bloom, and I found vanilla A instead. Moving on to the second set of experiments that I did, this was to determine the storage viability, and what I was trying to determine here was essentially what is best to store SPATs in and how long you can store SPATs for. So for this set of experiments, I made a total of 30 SPATs at one time, and for this experiment, I used the shorter production method for all the SPATs. And so 15 of these I stored in 100% Gen Pure, and then 15 of them I stored in 20% methanol. And then within those 15, I broke them down into triplicates that were stored for either 1 month, 2 months, 4 months, 6 months, or 12 months. And then also right before the time, 24 hours before the time point, I made a fresh set of SPATs to serve as the control. And so I deployed the 3 control SPATs, the 3 that were stored in methanol, and the 3 that were stored in Gen Pure on that project that I showed earlier, and deployed them here for a week in York River. Currently, I have the data for the 1 month, 2 month, and 4 months treatments, and I collected the 6 months, I processed them, and then the 12 month will be. And so from what I found from this was that essentially over 4 months, SPATs pick up just as much toxin as the control SPATs that are made within 4 hours of deployment. For the first 2 months, I did not find any asparasa 2. I only found that in the 4th month treatment that I did. And so shown in the two highlighted bars for the 2 months and 4 months for PTX2 and then the 4 months for acid 2, these fats that were stored in 20% Gen Pure picked up statistically significantly less toxin than the ones stored in Gen Pure when compared to the control. For everything else, they picked up just about the same. And I will not be around later, so if anyone has questions, feel free.
Thank you, Josh. We have about a minute if anyone has questions for John.
This storage is storage before deployment, so like you would versus,
Yeah,
okay, so. Are you making them, what are you doing moving forward? Like, are you gonna make them fresh since you make them within 2 hours?
I've just been making them like 5 at a time, because that's how much I like you can put in a beaker. But for like other projects and stuff, if you need a bunch, you can just make them all at once and like use them up at once together.
Okay,
So.
and in terms of storage afterwards, does that matter? Like, once they've been deployed, or you're gonna run them?
I just store them in the freezer.
Yeah, freezer.
Yeah.
Thanks.
And the containers I store in the fridge at 4 degrees.
And your control is 24 hours.
Yeah, I try to make them like as close as possible.
It looks like people are having trouble hearing online, so if speakers talk really loud and.
Okay, up next we'll have Vanessa Strohm, who is also from the Smith Lab, and her talk will be on comparison of intra and extracellular diarrhetic shellfish toxins and pectenotoxins in the York River from 2018 to 2020. Hello everyone, my name is Vanessa Strohm, and like Dr. said, I'm a master's student in Juliet Smith Lab here at VIMS, and today I'm going to be talking to you about some of the toxin samples we've been taking off the VIMS year from 2018 to 2022 with a focus on diarrhetic shellfish toxins and. So just a really brief overview, these toxins are produced by mixotrophic dinoflagellate dinophysis, which has been identified on all US coasts. And dinophysis is the causative organism of diarrhetic shellfish poisoning or DSP in humans. And there are 2 groups of toxins that these species produce. The first one are diarrhetic shellfish toxins, which include oxalic acid and dinophysis toxins. And these are the ones that pose the risk of human health. And then we also have pectenotoxins, which pose more of a risk to shellfish health. And here in the Bay, there have been no shellfish bed closures since the 2002 precautionary closure. But dinophysis has retained its presence here. So some recent research that has come out of our lab has shown cells as well as toxins present in the Bay, but well below regulatory limits. So moving into our sampling methods, first we deploy SPATTs, like Josh was saying, which are solid phase adsorption toxin packing discs. And we deploy these for 2 weeks at a time. The resin will adsorb a variety of different phytotoxins from the environment. We can bring that back into the lab, extract them, and then run them on our LCMS and look for a variety of different phytotoxins. And then we also have sieve samples. So this involves taking an 8-liter sample of surface water and concentrating it down with a 15 micron sieve to about 45 milliliters, and then extracting the cell pellet, and then we extract that pellet and run it on our LCMS. And for these 2 data sets, I compiled data from 2018 to 2022 with a focus on DSTs as well as PTX2. So getting into our data here, just to orient you first, this is the concentration of DSTs and PTX2 on SPATs, and this represents the dissolved or extracellular toxin. Here in this panel, each box represents a year. On the x-axis, we have the date. So I decided to focus on a time period between January 1st and July 1st, because this is when we typically see cell concentrations of dinophysis sort of increase in May. And then on the y-axis, we have the concentration in nanograms toxin per gram resin per day. And the red line here represents DSTs, and the blue line represents PTX2. So some trends that we're seeing for 2019, 2020, and 2021, we didn't really see any really large peaks of either of these toxin groups. They were just present in background concentrations throughout this time period.
However, for 2018, we see that peak in DSTs occurring in late January, early February, and then we see it steadily declining throughout the rest of the sampling period. And then kind of the same thing for 2022, the peak in toxin occurs a little bit later between February and March, and then there is a steady decrease throughout the rest of the sampling period. And then here is FIB samples, and this represents the intracellular or particulate toxin. And the panels are the same before, so we have each panel or each box is a year. On the x-axis we have date, and then on the y-axis we have the concentration in nanograms toxin for the year. So here we can see the first thing that stands out is that 2018 and 2019 look very similar, with that peak in PTX2 occurring right between May and June of both years. And then for 2020, we see something a little bit different with two peaks of the PTX2 occurring in early March, and then again in early April. For 2021, we see this peak in toxin occurring a little bit earlier in the year compared to 2018 and 2019, between about January to March. And then for 2022, we see two peaks of toxin again between February and March, and then again in April. So between these two plots, you might have noticed that there does seem to be a switch in toxin profile, with the SPATS having DSPs always being higher than PTX2, and the SIP sample PTX2 is always higher than DSPs. So for the intracellular or the SIP samples, this is typically what we see within cultures of adenoviruses, where PTX2 will be several orders of magnitude higher than DSP. For the SPATS, this is an active area of research in our lab. We kind of hypothesized that PTX2 might be less stable in the water column, so it might be undergoing degradation, or it could be sorting to particles, and that's where we're going to up. So with that, I'd like to thank the Smith Lab, Juliet, Marta, and Nour, Josh, and two previous students, Sarah and Michelle.
All right, next and last from the Smith Lab lightning talks, we have Nour Ayache, who is an assistant scientist in the lab, and she'll be talking to you about the effect of low level exposure to HAB toxins on the larval life stages of eastern oyster.
Good morning, everyone. So my name is Nour Ayache. I'm a assistant research scientist here at VIMS, working with Dr. Leah Anne Gibala-Smith. And today my presentation is going to focus on the effects of low-level exposure of some HAB toxins like PTX2, toxins, and Crassostrea on the early life stages of the infant oyster Crassostrea virginica. So just for a brief introduction, recently there have been some unexplained larval mortality and hatchery failures. And these mortalities have been occurring at the same time with seasonal abundance peak of multiple toxigenic HAB species like Diarrhetic, Egerton, and Phytoplankton. In addition, really recent studies have shown the infiltration of the detection of low levels of PTX2, toxins, and Extracellular in the post-treated hatchery water on both coasts of the US, here in some hatcheries here in Virginia, but also in Washington state. And this water is usually used for the culture of larvae. And when we look at the literature, there's really little to nothing known about the effect of these low levels of toxins on the early life stages of these species. The only two studies looked at the impact of PTX2. And for example, we see with Gaillard's study, they showed the decrease in the fertilization success of the oyster gamete after 2 hours of exposure to low levels of PTX2, around 5 nanomolars. And in Pectenotoxins study, our colleague Sarah showed a 50% increase in mortality of oyster larvae and the larvae after 48-hour exposure. But this time they used high concentration of purified PTX, concentration of 223 nanomolars. So we decided to continue this studies, but this time there's really low concentrations of PTX3 toxins and Extracellular. And this concentration was true because they are ecologically relevant, and they represent concentration that were found in food concentration, for example, of Diarrhetic that were found here in the bay, or concentration that were found on SPATT extract for Extracellular, or for Diarrhetic, it was its concentration representative of the one that were found in water samples on computed sounding Washington State. And we exposed the larvae, 7-day larvae to these toxins, and we added also two controls, one containing 0.2% of methanol. And after 48 hours of exposure, we monitored the larval mortality by microscopic observations and the toxin accumulation in the larvae by SCM estimates. So these the primary result. This graph represents the percentage of mortality of larvae with the different types of toxins that were used. And first of all, we see a significant increase in mortality relative to the control, both controls, for Toxin and PPS2 and Azaspiracid 1, going from 7 to 11%. But also we saw full immobilization of the larvae after 48 hours of exposure and toxin accumulation in the larvae. This group. Here we can see some larvae that are dead and other that started to get immobilized, and after 48 hours all the larvae were immobilized. So these were just a preliminary experiment, and our next steps are the most important. So we're aiming to collaborate with other hatcheries, especially here in Virginia, and identify the correlation between the presence of this low concentration of toxins on the and the effect on the early life stages of different life stages of oysters. And then conduct a range finding experiment in the lab using the concentrations that were measured in the water sample in these hatcheries, and then determine the difference and the lower toxicity threshold at which no mortality is detected in the oyster, and also determine the impact of the mix of these toxins on the early life stage. So these results will help us provide options for shellfish growers and to mitigate the effect of these toxins and for example, enhance the filtration system in aquaculture facilities. Thank you so much.
We do have an online question for
I'm about to leave, but I can answer it.
okay, before you leave, right. Would it be possible, or how might one extend this work to see how storage might affect how SPATT concentrations might predict actual water concentration?
Yeah, because the SPATT kind of part, you can't really determine the actual water concentration from SPATT resin. I also take like a whole water sample when I do my sampling, which gives me like an actual concentration, but normally toxins are very low, so it's hard to do like that. But I guess you could kind of like compare the two data if you continuously put water samples to see how it compares to SPATT data.
Thank you. Okay, up next, we have Margaret Mulholland's talk. Margaret is a professor in oceanography at Old Dominion University. She is a biogeochemist and leads the water column biogeochemistry group, as well as the Phytoplankton Analysis Laboratory in the Department of Ocean and Earth Sciences. And she has worked on harmful algal blooms for over 30 years.
Yeah, we took the A out of it, so now it's not Earth, Wind, and Fire anymore. But this was actually a picture, a color enhanced picture that a former student, Brian Morse, took during Margaret. Yeah, I love that picture. But essentially what I'm going to do today is give you an overview of the shellfish monitoring samples that we collected this year, the bloom response done by ODU, BVDH, and talk about the notable 2022 blooms. Heads up, there really weren't huge. It's a pretty light year. Pretty light year, but there were things. And so I'll talk about the monitoring results, some related projects, and our New Year's resolution. I always like to have resolutions for the next year. Yeah. So for in terms of VDH monitoring, what we get is there are monthly collections for routine fixed sites for the Division of Shellfish Safety monitoring. And we get bluegill preserved samples for those, and we get unpreserved frozen samples as well for live screening for toxins. In 2022, we got 467 FYI. And we also get bloom response samples Although the ones from the estuary really aren't as abundant as the ones we get from freshwater. It would be nice to have better surveillance of some of the saltier waters. But we don't get as many responses for those, I think because there aren't as many people that have their eyes on it. So, the bloom samples, we get the response to bloom reports. We only had 4 bloom samples and 7 more that were likely blooms from the estuary, from the salty water. So that is truly under surveillance, I think. Okay, so where are these samples from? This just shows in dots, colored dots, where the Bay program monitoring samples come from, where those stations are. And those are just monthly samples. And so we really rarely detect blooms in those because they're so infrequent and they're fixed stations. So if you're going for that GPS coordinates, you're not, sometimes for blooms, it matters which side of the boat you sample out of. And so, so they are very unlikely, but we get 7 Chesapeake Bay monthly samples year round and 7 tidal tributary samples monthly. And I have the January to December highlighted because they did stop collecting samples and the trips for us in the winter, but I had them started up again because we were working on a paper on the winter time in the Chesapeake Bay. And in fact, there are and heterocapsid. Yeah, in blue, so are those Bay program monitoring ones. Yellow are the DSS stations. There's 69 stations that are monitored again monthly, but year round as well. And then there are some CBE toxin samples, but we don't get many of those. We only get 6 of them. So we don't get a lot of those. And then we have a time series site in the Lafayette River where we do bloom detection and early detection of Margaret in particular. The HRSD, the Hampton Roads Sanitation District, does data flow mapping on the James River, and they reduced this to being bi-weekly this past year instead of weekly. And they collect samples for us when they see this colored water, but since COVID, they sort of interrupted that procedure, and so they haven't really gotten back and they haven't been collecting as much water as we'd like for those samples. And again, then there's HAB response that just randomly. Okay, so I think I already even said a lot of this, but the HRSD, the data flow, like I said, is now every 2 weeks, but they do multiple segments of the James River, and they use chlorophyll of I think it's 15 micrograms per liter to trigger them to collect samples for us for taxonomy. But we didn't get a whole lot of samples this year. Like I said, since COVID, they've been out of the habit. We did get Bay programs year round, and we observed heterocapsid blooms in 2020, which is why we had this happen again. And while we don't have much data on it for 2022, we've already had heterocapsid blooming in 2023. So it's already happened. And we continued our 3 times per week sampling at the Lafayette River at our time series site there. And we also have been starting to make a flow cam library. Because we were able to purchase and install a flow cam this year. And we've actually worked with FlowCam to have their proprietary software be less proprietary. So that we can actually download the images and put them up into EcoTax or any other platform that we choose to use in the future. So. Okay, so there were blooms in 2022, but they really weren't by most of our surveillance techniques. But we did have reports about this colored water here, there, and everywhere, but we didn't pick them up well in satellites even. So, so we seem to be missing something. We did observe blooms at our time series site. We're still counting the samples, but I don't have those data to show you, but they were there. And we have reports of them being transported around a little bit. But again, it doesn't show up in some of the surveillance techniques that are broad scale, like the satellites. So, yeah, so we need to figure out ways to better surveil, especially in our estuary waters where people aren't looking at them all the time. This is just showing the normal bloom progression that we had. So I just wanted to show you, we usually get a series of blooms starting with heterocapsid in the wintertime when the temperatures are low. And then we usually get for a central minimum, we can get dinophysis, pseudonychia, Gymnodinium species, then Carlodinium, Strixella, Akashiwo, Margolopidinium, and Alexandria. So we have our little parade of blooms year round. And this is, we published this in 2018, this figure essentially, but it's the figure we published is on the left and the data that it was derived from the company. Okay, so what happened in 2022? Well, we did have a little bit of heterocapsa that came up in March-ish and some Porocentrum in the late April, early May. And then we did get Margolopidinium observed in samples in the summer. And we know it was there, but like I said, we didn't get a huge number of samples with it in there, other than at our time series site. And that's to show on the lower left is 2022, and just for comparison, 2020 and 2021. So, while it seemed light-ish, we still had a few things that popped up. Okay, some of our less abundant species in, oh, sorry, the legend got messed up. I couldn't get it to close the gap in Excel. I don't know if I need to update the software or what, but it's just driving me nuts. But this is just showing Dinophysis, Pseudonychia, and Carlodinium in 2020 on the left. And then I have 2022 for a comparison. We added a couple species to this as well because we actually had the Corenia in a couple samples, which I don't know for those of you familiar with the Gulf of Mexico, but Corenia brevis is bad news. And if that's introduced into the bay, we could be in deep doo. And but we did have some Carlodinium pits, and we noticed the scale is a little off, but again, these are still pretty rare species in the bay. And then afterwards, we have the pre-nest that and just showing in the Pseudonychia, in general, there were low cell densities, but a pretty widespread distribution in the bay. And so the light blue on this graph on the right is the 2007 to 2021, and dark blue is 2022. That's just so all these maps will look sort of the same. But we don't have them for every species here. It wasn't worth to make it. Again, here we have 2021 data, 2022 data, and again you can see they were a little more abundant, but not very abundant in 2022 compared to other years. And you can see the figure on the left in the dark orange is the 2022 data. The lighter yellow is the compiled 2007 to 2022. So that's Dinophysis. Did I say I used to. I did. Okay. Yeah, okay, this is just showing heterocapsa in its distribution 2022. Even though, you know, we don't have a big data over there in 2022, we did have a couple samples with large populations, but just a couple. Yeah. And then Porocentrum, oh, yeah, I did, this is just, that's the old figure on the left, the figure from 2022 on the right. And again, it was widely distributed, but not a lot of them, except for in very few samples that were collected. And Carlodinium, this is the 2022 concentrations, again, widely distributed, but not really huge population. So generally low in abundance, but they were there. Margolopidinium. Now we have the we didn't make a new figure for 2022 because most of the samples were pretty low. But like I said, we actually did have some bursts in our population and lots of observations that they were there, and we measured them at our time series again, and we saw them in a lot of places, but they just weren't reported or not sampled. So not a huge amount of data. Then I wanted to show you the satellites data because the satellites weren't a huge help this year. We didn't see a lot. You can see some populations here that these are from 9/20 on the right for September, and that's Alexandrium, and you can see it up in New York there, and a little bit in the lower James River. And on the left is from July, and you can see some marginal epidemic down there in the lower bay. And so we had it, we can see it sometimes, but a lot of these, I really had to scour the satellite images to get something to show up.
It was actually a mix of March and now.
It was Margaret, yeah. And then it's hard to get out to the main stem of the bay to figure out what's that in the middle of the bay, that block. Yeah. So for Alexandrium, again, we didn't make a new figure, but so this is 2020 data, so it's full, not really 2022. But again, we didn't have high concentrations here. All the things that were reported that we had samples for were low, even though we know there was a big population that hung out in North for a while. and it made its way into the James River, but it was really under surveillance. So now I want to switch gears a little bit. I talked about the
Get
IS.
the audio on my computer. So I'm called in.
Someone is unmuted on the remote.
Maybe you can mute. You can, I think you can move. Okay, it's all the way over on the screen for.
I'll be.
Yeah, so we, like I said, we were able to purchase and install a flow cam and then a carbon get OU. So we played, she has an IMCP as well that we had deployed and have been running samples with. It wasn't available this year because it was out to sea, so we didn't use it a lot. But the flow cam, we've been running samples through both preserved and unpreserved. This year, our time series samples, we're even doing Bay program samples. And we're trying to work with Maryland and look to work with them to get a Bay-wide sort of image library of all the species that occur there. And we're hoping that, that we can increase our sample throughput using these. We still lack labor to train these image libraries and curate them. And so it'd be nice if we could do that. The other thing we've done is we're working with Americans a little bit. If they're interested in hosting a Mid-Atlantic image platform for us to use. And so we're hoping we can develop that down the line. And then we submitted a HAB proposal with VIMS to do a better job of surveilling and transitioning forecasting systems management for March and Alex at least. And a large part of that is going to be building low-cost imaging systems called microscopes that we hope to deploy widely with samplers in the field that they can put we can build them for about $1,000 now and deploy them with people in the field and then keep everything in the water and upload them to our platform and have almost real-time data. We can get it training and working. So we're hopeful. And this is just a product showing a bunch of Marge in an image that's been in a library that's been curated. You can see there's still a couple things that don't belong in there. Yeah, so I mean, it's a process of machine learning that you train it, run it, retrain it, run it, retrain it in an iterative process. So, but we have students working on different projects related to chain length, swimming speed, and all this stuff that we can now get through images that we're able to go through. And we're learning a lot about different life stages as well of different things. So that's been really great. Okay, so how did we do with our 2022 plans? Well, we continued our merging and sampling of databases, so we continued that. We somewhat resumed our HRSD sampling after COVID, but it wasn't really successful this year, so we're going to really push it this next year. We still did our time series sampling at the Lafayette River, but that's a challenge. I have a lot of students graduating this year, so we have to abandon the time series station. We want to. We've already established FlowCam and are working with them and work with them. So now we have a downloadable image database from FlowCam. It's not proprietary anymore. So we did that. We're still building off multiple programs, piecemeal funding to try to get better surveillance. Hopefully we can get this HAB thing funded so we can have some targeted resources to do the image library curation. And we do have a lot more student involvement. We've had one of the undergraduates this year today, Gabby, in the back. And we were able to employ 3 undergraduates to do different projects with some of the databases. So we're really happy with that. They've been really really successful. And so we have lots of manuscripts still in preparation. And our New Year's resolution, we want to keep working on submitting some of the overdue manuscripts that we have from all this sampling. We want to do more year-round sampling because of the blooms in the winter that happened, and I think we have some of that lined up with undergraduate students, and I've actually dedicated a class to going out weekly on the Prep series. So our oceanography laboratory is doing that. We're working on laboratory experiments, culture experiments still to do growth kinetics and try to better understand why and when blooms are occurring. And we're always trying to find more funding to tie our research in with management priorities. And we're gonna work with we hope to do more SPATTs with colleagues here in the Potomac and Lake Gaston, and we've done some work before as well. And so hopefully we'll be able to do that, and that's all I have for you.
How do you know which?
It was not Revis that we found.
Okay.
No, it was not Revis. I think, which one was it?
So we for sure had Perennia Apolloniaceae,
Yeah.
which forgive my pronunciation. Pronunciation, I guess. But they had another cell that was very like cell-formous, like not really make modi. Haven't decided what to call it.
We had something in the door.
We have one Perennia cell. We didn't know which one.
Yeah, we put it there.
Also, Rick's not tested. He said there are two different satellite products. One shows that a 0-7-20 blooms much better than the other. So that is.
Okay, thanks. That we show me which product that is. Yeah. Thanks, sir. Thank you. Thank you. Looks like we're.
Okay, up next, we'll have Todd, who will give the BDH marine biotoxin recap 2022. Todd has worked with BDH and the marine as a marine science supervisor for 7 years. He is a member of the ISSC Biotoxin Committee and has worked with harmful algal blooms and phytoplankton monitoring in the Chesapeake Bay since 2001.
Thank you. Yes. Okay. Everybody. Well, thank you very much. I think this is going to really play well into following up Margie's talk. So we're going to talk about a lot of the same sort of blooms, but from a very public health standpoint. So, like Danielle mentioned, we are the public health agency, so our primary concern are marine biotoxins that can make their way to cause human health concerns through shellfish poisoning. So within the National Shellfish Sanitation Program, there's a list of biotoxins, paralytic shellfish poisoning, amnesic shellfish poisoning, neurotoxic, diarrhetic, and exosporaphin shellfish poisoning. So all of these we have included in our marine biotoxin plan. I should say a lot of these slides might look familiar, especially if you've come to these for prior years. Throughout all of them, I've tried to highlight new things this year in either orange or red. So those would be the kind of highlights to look at. Like Margie mentioned, this is a multiple programs all combined into one. So we have lots of different metrics. We have the Lugol samples that Margie talked about that we collect and go to ODU. We have those that are streamed in our shellfish offices. So again, thank you to our shellfish staff that are field staff that are collecting those, and our lab staff that are doing some of the first screening on those. And then we're collecting spam calls in the very. We're collecting now multiple samples as well. So we're collecting frozen samples at the point of the collection. in the field that we can run ELISAs on. One of the just practical things this year, I think we're gonna move from these 50 ml tubes down to a 15 ml tube. Still enough water, we can freeze it a little faster, pre-thaw it after, it takes up a little less space. So that's, minor, but we'll be rolling that out next year. And then a few years ago, we added these PCR samples, so this is working with VIMS, where we're taking now a collection at every single one of those stations that Margie showed, and on another map, as well, that are being screened using qPCR. So we have the morphology, we have toxins, we have qPCR, and then we have these bloom response samples as well. And then now for the last several years, we've expanded more with oysters. So we're tasked with looking for public health concerns of toxin in oysters, and now we're, besides taking all of these water samples, we're also looking at oysters directly. So we'll talk a little bit more about those. Again, there's lots of algae in the bay. There's over 1,000 different types of species of algae in the bay, and relatively few number of those are ones of concern. These are sort of a list of all of the toxin producing, biotoxin forming, poisoning biotoxins within the model ordinance. Again, some of these, we're fortunate enough to say that within the bay, we don't have the saxitoxins producing Alexandrium for one, Biotoxin for another. So the Corenia that we've seen periodically, so far we haven't seen Corenia brevis. When we've tested for Biotoxin, we haven't identified that. So hopefully we'll continue that to be the case. The ones we are more concerned with are the Dinophysis and the Pseudonycium. So I'll focus more on those as well. And then Azotoraptis. Here,
good morning, happy Friday. Am I calling you at a bad time? I can call you back later. Yeah, go ahead, just call me back after that. It's all good. Yeah, so I think we got it in 15 minutes. All right, bye. Thank you.
If all these are.
Yeah. So I don't know where we were, but we're going to talk about it a little bit more. They're a newer one to us that came out of the CB projects. They're relatively newer, not just within our region, but throughout all of biotoxins. It's a relatively newer species, but it has been identified now within Chesapeake Bay. Again, some of the things we're doing more of now, we're doing more of this expanded routine seawater toxins. In the past, we were really doing toxins only after we were seeing cells in the high cell counts, then we were following up with toxins. We've expanded that, so we're doing more of those routinely. And again, we're doing more shellfish deployments at more sites. So this looks very familiar, I think, as well. Again, we have multiple programs going on. Margie already covered the Bay program. Glad to hear that this is now year-round. The shellfish sites here in yellow are the ones that our VDH field staff are going out to. And then the CB Tox ones, these pink sites, were again this collaboration between VDH and VIMS over the last now several years. And that started off as a EcoHab funded project. And then we continued it on sort of with more collaboration just between VDH and VIMS in subsequent years. So we had 12 original sites and then 4 sites where we had shellfish, and then we had 7 sites. And then this is the current app now, we have 8 sites where we have shellfish deployed. So this is, we've added, especially the Chincoteague site now in this last year, we've seen high cell counts up there, but we've never had any oyster data. So again, thanks to all of the staff that are working on these, especially Jackie and Evan, you're here. These are really the ones that they've been working on. So there's, again, Margie mentioned, there's other routine monitoring or other monitoring that's going on outside of this as well. So I'll start with the azosparachids. So azosparachid toxin can be produced by Azodinium and Anthedoma. These cells are very, very small. They also kind of look like heterocapsa and a handful of other things, and really aren't something you can monitor for using light microscopy. So we worked with Kim and Gail and her lab, and they've been now running molecular analyses on these for the last several years. And this is really our only tool. There's also not azosparachid kit. So for some of these other ones, we can go out and purchase ELISA kits for thymoic acid, for saxitoxin, et cetera, but not for azosparachid. So it's something, it's not great. We have it in the model ordinance. It's a toxin we need to manage, but there's not a approved method for it. So right now, the molecular screening that we're doing with VIMS is our tool that we're using. And then we would work again with VIMS to do LCMS if we needed to. But for the most part, we've seen that these cells are out there. They're kind of spread everywhere and have relatively low, or I would say low densities. So we don't have anything. I think everything now was less than 30 cells per mil. But we're seeing some detection, and we can talk about like where we want to put that lower limit, but somewhere between 0.1, above that, and 35% of our samples, had these Pectenotoxins. I'm just going to try to avoid saying that, but I think that's
I'm glad you said it.
so I think in terms of moving forward, this is one where we're kind of being aware. We're continuing to surveil this, and I look for this using the qPCR method. Dinophysis. So again, this is another species that we do see. We know is out there. We've detected the toxin through the SPATT work, through the CD Tox project. We do see these cells that are everywhere, and we've done a greatly expanded toxin screening on water samples from these. And I think similar to what Vanessa was seeing in their SIB samples, and these are smaller, but we're still not detecting any of the toxins. And so I should say this is the oxalic acid and the DSTs. So it's not the PTX2s, but it would be picked up for we did the hydrolysis step. So it would pick up the DTSs. Is that right? DSTs? DSTs. But so even at cells, cell densities that in Texas, maybe or other places have been reported to produce toxins, above 5. We're not seeing toxin in these waters, which is good. So if we are concerned about these, I feel like we need to do some more work with SPATs in terms of we're not seeing the toxins in here with these tools, which is good. It's not a bad thing. But I think SPATs would be the way to go, for doing more work with this toxin. Different story here now with Pseudonecia. So I'm going to spend most of the time here with ASP. So Pseudonecia diatom can produce thymoic acid associated with amnesic poisoning. And again, like these other ones, we have a widespread distribution. These blue dots I'm seeing from the back aren't the easiest to see, but the fact that you can see them everywhere is important. We see Pseudonecia throughout the entire area, both on the seaside and within the bay. And what I wanted to point out with this figure, so there's lots of different species of Pseudonecia. And so Pseudonecia, even cryptic species. So there's a lot that are very hard to identify and differentiate. Definitely with light microscopy, but even with electron microscopy, they're pretty tricky. But what we've done both in Virginia and kind of, I think, as a whole across the country, sort of try to look at breaking these down for a screening purpose into larger and smaller cells. So the larger cells, kind of what we're saying is this 5 micron width, the width of the cells, not the length of the cells, are more associated with thymoic acid producing species like Pseudonecia australis. I think that's kind of like the poster child of DA producing Pseudonecia. And so we have those, and then the smaller ones are less commonly producing toxin or producing less toxin. And this sort of breaks down like where we're seeing these big cells, these dark ones, and the smaller cells. And it's messy, we have, but mostly like bigger cells in the ocean and smaller cells in the bay. It's more or less what we're seeing. So now this is toxins, so we're switching to toxins, and these you really can't see, but we ran 132 samples this last year for the MOCA. So this is a lot more than we've run it in past years. About half of those, we did get detectable domoic acid. These are from seawater samples. So if there's these little blue dots that are all throughout here, these are all negative. So just about everything within the bay where we're seeing these smaller cells, they're toxic. And every site on our Atlantic, on the coastal eastern shore, was positive. So if it's green, it was above the detection limit. If it's yellow here, I have it above 1 part per billion in the seawater. So it makes sense. I mean, we have multiple strains going on, multiple species. Even within this is another one of those frustrating algae species where even within the same species, toxin production can be associated with other environmental factors, so nutrient on demand. So there's a lot going on. Usually it's if there's limited nutrients, there can be higher domoic acid. So I think it's probably a multiple combination of different species composition out in the ocean. And then you also have the possibility of different environmental conditions as well. These figures here are relating, again, this cell counts, which is here. So the cell counts to domoic acid production. And out on the seaside, for the most part, where you're seeing cells, we're getting some sort of measurement of domoic acid. But I'm still getting a lot of DA here when I'm not seeing cells. So when I'm at my detection limit for those counts. Within the bay, it's kind of a mess. Like we have a lot of cells and no toxin, but then we still have some toxin where we're not seeing cells. So this is something I really am concerned with and want to work more on in the coming years. So using different ways to kill the cells, different ways to measure toxins, things like that. Yeah, exactly. So I think that's definitely one of the ones I think we can talk through. I know Gail is talking about there's some new molecular fibers. Okay, so I won't spoil that. So these concentrations that we have seen in the seawater this year are consistent with what we've seen in the past year. Not that these are really higher. We're in the same one to just around less than 2 parts per billion in the seawater. So all of this is seawater. So what about the oysters? So we put out oysters last winter at 7 sites and sampled them through February. All of those were below the detection limit for domoic acid in the shellfish meat. And then we put them out in the summer again and sampled them from July to August at 5 sites. And of those, 1 sample came back positive for DA, and that was from the James River. And that sample was at 0.356 parts per million. So this is within the oyster meat. So this one we have a closure criteria for, which is 20. So we're almost 2 orders of magnitude below that closure criteria. And this concentration here is also consistent with the highest level. So in the still kind of in the press, but in the CB Tox shellfish study that our pieces work, the highest level that they saw within that was 0.579. So we're, I think, pretty consistent in terms of the range that we're seeing from these samples. And again, still well below that closure criteria. So that was in the summer. So we've got oysters out again now, and we're going to try to keep those out and do a rotation throughout the whole year. So we're aiming to have oysters out that we can sample ongoing throughout the year. That's the goal. And so we again have them out there. There's some right here at the VIMS here as well. So those are in process. I should also say this is the ASP data. I didn't make them out for the BSP. The BSP for all the samples we've gone through, and we haven't finished all of them yet, but all of those have also been BDL for the BSPs as well. Briefly, I wanted to talk about the flow through. So last year, I think I presented, we had gotten a grant from the ISSD, which involved VIMS as well, to kind of develop this tool. We got some equipment money to buy these sods, and we rigged them up very similar. You might even say stolen from the data flow system that VIMS has, where we're pulling water on board. And then measuring the chlorophyll and other parameters. And then we're also passing that water through a chamber where we have a SPATT. So the idea is that we could measure toxins while we were underway. Of course, when we used it, there really wasn't a whole lot of blooms in those years, since we didn't have a lot of data. And what it did show was that this is going to be a much stronger tool when we have high biomass blooms. And so we did come out this year and see, or last fall, and go to the Margaret bloom in the lower York. So this was our cruise here. The fluorescence is the underway chlorophyll, and the range was huge. So from less than 1 to over 700 micrograms per liter. And the really cool thing, so we got some samples and that corresponded with some high levels of large and lower levels in Alexandria. But this was the same day that we were out there with the satellite data. And so this is the September 2nd. And so you can see what we saw here really matches up well with this remote sensing data. So we were impressed with that. And this is also sort of what it looked like in the field. So again, look at the difference here. We've got 700 micrograms per liter here and almost nothing on the other side of the river in the same area. And this is satellite data from this year as well. And I would say, again, like in terms of the satellite data and our use of it, this continues to be very useful and important for us. There really wasn't, like Margie was saying, there wasn't a big fall bloom in the lower part of the bay, in the James River, but we did have this pretty dense, very dense bloom concentrated in New York. So, and it was also helpful. There was transport of that bloom across the eastern shore, and so we had reports coming in from the shore saying what's going on over here. And we were able to look at the satellite data and say, well, it looks like we're seeing a continuation. And then when they collected samples there based on, again, the satellite data, we confirmed that this was large being transported across the bay. So we continue to use the NCOS satellite data. It was, again, I think I mentioned it last year, but this was really helpful during COVID. We had just a few months that we were home. And during that time, we could even look at the website, see if there was a bloom going out and send additional field crews out to there. So, so that's been helpful. And then last slide, what are we working on now? Where are we going? Again, the biggest thing we're looking at these sites. We're looking at year-round studies. I think putting out more, doing more toxin testing, moving more towards toxin testing, and figuring out what's going on with these relationships. So we can now run toxins at OU with the CAS cube there. We have plate readers in 2 of our field offices, so working to get everyone trained on those as well. And then I really am looking forward to working, Margie and Kim in the next couple of years, trying to figure out a better, again, relationship between this toxin, especially with gas. My focus. They're looking to understand that relationship. And then I have to just have up here briefly cyanotoxins. So I don't have much cyanotoxin data here. Something that I think is coming down the pipeline in the ISSC. There's a lot of talk about looking into it. We've been working with Wade up at the FDA. So he has some samples as well that he's looking for microcystins in some of these. And so we're gonna make sure that we're not behind the curve on looking at what the potential is for cyanotoxins in shellfish areas. There's been a lot of meetings recently that have included a lot of people here as well. We presented this work at the US HAB Symposium last fall, and there was a meeting here last month that was really good that Noah put on, that VIMS hosted, the Sea Grant workshop that some of you were involved in as well. And it was about talking about with the industry, so not just us as regulators, but the shellfish industry as well. Not just shellfish, there's recreational fishermen here, and hatchery folks, about using remote sensing and talking about HABs and how to make some of those tools better moving forward. We're still working with DEQ. We're looking forward to finalizing our MOA with them on HABs, and we'll be heading down to the ISSC in a few weeks. I'm on the Biotoxin committee there, so we're moving some changes forward there as well. But I think that's everything. Thank you. Yes.
Do you track shed numbers of sulfa or mesodinium? Shed might be a biotoxin producer and mesodinium might be a precursor.
Right, so, yes, they just left the room, I think, but, the ODU lab is, we're looking for it too, and both, VIMS and ODU will identify, the phytophytes, and we've talked about, chattanel as a possible biotoxin producer in the past. We've done a couple spot studies here and there where we've seen some, and I haven't been able to ever see biotoxin in those samples. So I know you've got it. But and then we also, there is measurements of mesodinium. We've talked about that as well because Lyanophysis is such a low biomass, we're not gonna ever see like a chlorophyll signature for that, but there is a chance that we could see, again, mesodinium as a precursor, or even stepping one level back, cryptophytes as a mesodinium precursor, and then as Lyanophysis. Yes. Dr. Jones.
Thank you. I've had experience with microcystic blooms and atomic back in the 80s and 90s. And so with microcystis, we were always seemed like it was not going to survive in the same place as oysters.
Right.
So are there other cyanobacteria that are toxin producing that might grow in an oyster?
So that's kind of been our thought as well, is that, like freshwater is a freshwater thing and oysters are an oyster thing, and we have that salinity. So I think microcystis is, and microcystin is still one of the toxin microcystin, but that species as well, I think, is one of the ones that we still would be concerned with. But I know from, I don't know if it'll be good, but like there had been microcystis blooms even in Colonial Beach, and so that has kind of, in the early 2000s, that kind of where that overlap is. In the James River, we've seen some microcystin transport down into the mesohaline portion, where again, we're getting into it. The other part that I hadn't initially considered, I was thinking the same thing. We've got this big freshwater river going down, but we also have all of these smaller trips and even ponds that have, farm ponds or other ponds along the way. Yeah, York River State Park, where you can have a bloom right up against that river where it would strong salinity gradient. We know that there's aquaculture going on up there. So that would be another potential where you might see a much more localized impact of a bloom coming on. And my guess is that we're going to see something more like that. So there could also be other species that are producing toxins, but yeah.
So if they were like freshwater here and extreme water here, and there was a lot going on here, and then they bled over and they started dying because it's all water, they might.
Exactly. So the fact that like the cells don't survive doesn't negate that there could still be a health risk there, because once those cells lyse, like that out of the water. I don't know how that would work in terms of, if the oysters are getting primarily, if they're ingesting the particles, if they're ingesting the cells, I'm not sure. Yeah, so if the cells are lysed, I don't know what that would be for the shellfish. So that would be my only question. It'd be a good study to look at, like, does toxin make its way into the oysters? Maybe there's been stuff there if you have life solubles.
We've seen the DNA.
Yeah.
But Sarah has picked it up. SPATT.
Right.
But I don't.
But
Did she do oysters? Margaret? Did Sarah pick up microcystin in oysters? I know it was.
So there was some,
She saw the oysters.
Yeah, and then that wasn't identified, I think, on the David and the James, I guess. But I think in general, like the idea that toxins are primarily making their way in as particulates.
I think there's going to be an interesting presentation of the symposium that I have this year. That's going to be talking about microcystin and oysters and Vibrio. You know, like between those three. That'll be some of the forest in that moment.
Yeah, I don't see it. I don't think the science is there now that would put microsystems as a regulatory criteria. But, I mean, Amy's here, we've looked at the toxins, that were identified within the oysters and tried to look at all of the science that's out there. And what we've seen is that even the high concentrations that we've identified in the Virginia oysters are still well below what's been classified as a health risk elsewhere. Is that still correct? Somewhere in that, looking at the California health risks and Australia, you know, we were still below those.
Happy had a comment for scenario. So she said California and other states through Shellfish and SPATT documented that microcystin can travel miles downstream of 20 miles. And Maryland has also seen microcystin production by Pectenotoxins, which is an iris.
Right, yeah, as a Phytoplankton. And that's one, again, that would be, we'd be working again to merge sort of programs together, there's Phytoplankton monitoring kind of through the Bay program, but that's not something we're doing as a monitoring.
I have just one question. When you found the deploying acid in shellfish, did you collect it at the same time as when you were seeing cells in the water? Or toxin on spats?
No, I don't think, I think that there's still a disconnect that we're not, so we only had the one that was in there, and ironically, that was from our first sample, so we're getting oysters from the James to dredge, we're dredging them for color rocket and bringing them out, and of course, our control one is the one that came back as positive, so that wasn't even one of our studies. We have a James study site, but that was from there.
Thank you, Todd. There'll be a question and answer section after this next call, so we can save any questions. Okay, next, Kim Reece doing the VIMS, as part of VIMS and is the marine lab recap. Kim has been at VIMS for 28 years and has served on the HAB task force for more than 20 years. During this time, during her lab members have been working with the Department of Health to monitor lower Chesapeake Bay water HABs for HAB organisms using microscopy and PCR assays. Her lab also conducts molecular genetic studies on aquaculture shellfish species and aquatic pathogens of humans and shellfish. The primary focus of her research is examining the biological impacts of harmful algae on aquatic organisms. The key component is the development and optimization of molecular diagnostic assays for viral, bacterial, and persistent organisms, including the HAB species, in various environmental matrices, including water, sediments, and shellfish.
Okay, that's a little long, but. We're gonna talk about qPCRS. Todd kind of introduced you a little bit to the qPCRS. So this is gonna kind of build on what Margie's been saying and what Todd's been saying. And talk a little bit about, so this kind of is 2022 blooms. And as Margie said, this is a little bit in quotation mark blooms. We did see some heterocapsa species blooming here and there. Akashiwo, yeah, maybe some blooms here and there. Probocentrum, Mycans cordatum, which I still call minimum, sorry, I might sneak that in here and there because I haven't gotten used to the new name yet. I finally got Margaret-Lepidinium, I think, I don't call it cockroach as much. So mid-spring in the RAP, we got a couple of samples with high levels of Heterotaxa rotundata, maybe almost to bloom levels. Carlo was not at bloom levels, at least not for Carlo. We saw some that had maybe tens, maybe 50 to 100 cells per mil of Carlo. Not anything close to what you would call a bloom level of Carlo. We had some P-min, Probocentrum cordatum. The York River, we again had Heterocapsa, mainly Triquitra,
or Quetra, however you want to say it, and Pro in the spring. A lot of mixed samples this year. I don't know if that's what you were seeing down at ODU, but we saw these samples that if they would come in with discolored water, they were just a mixture of a lot of different species. So even though they were a mixture that none of this individual species might not meet that criteria of 1,000 cells per mil, if we would call a bloom, but it was a mixture that might total 1,000 cells per mil with a mixture of heterocapsa and Coro and Carlo, but nothing. In June, we did get some, I haven't gotten used to Cotylum gyrodinium and striatum lavenderina. Where's Kathy? I'm sure she can pronounce this better than I do, but anyway, Leah, you can probably.
I'm the worst.
Anyway, and with some chlorophytes and other things thrown in there, we just had these mixed blooms of things. July, we finally got, this is not the best image. There was a better image, I think that we got. This was the one that was on an email attachment. I pulled it down last night. We had a bloom of Proro and Lavenderina and Scripsiella, and down in New York, it was like this mixed bloom of heterocapsa, and we had Rotundata. It kind of switched from Petra to Rotundata down here in New York. We had Proro. I put the bottom, it was minimum and Lycans mixed together. We had both Proros. We had some Carlo thrown in there, Acachivo, and Gyrodinium or Lavenderina. And switching over right at the end of July, we were switching over to that Margaret. So March was starting to creep in there right at the end of July, beginning of August. And I looked at our little board. Savannah Mapes, a graduate student, she writes down, starting what we call bloom mania. And bloom mania is when Margalepidinium and Alexandrium started blooming in the New York River. And our bloom mania started with the Margalepidinium bloom right about the beginning of August. And she had it tracked all the way until the end of September. So March in New York River, along with Alex and Proro was in there, all the way through August until almost the end of September. I think it was September 27th. We kind of had the end of that. So we had a very long period of bloom, but it wasn't, there were some very dense bloom patches here in the New York River. I mean, it was really long. And we had Alex patches, okay? So Alex was thrown in there this year. And a lot of those samples we got were mixtures of Margalepidinium and Alexandrium marlata, along with Prorocentrum thrown in there. Every so often you get some Akashimo thrown in there. It was just the strangest year of the. So these, again, shout out to Todd for all of these wonderful maps of you. And these are specifically the qPCR results, okay? So they didn't always correspond with the visual results. The samples are replicate samples. Okay, so they samples taken for visual and a sample is taken for qPCR. Okay, this is Carlo, which is very comparable to what was seen in the visuals. Very low levels of Carlo by qPCR and by visuals, but it was everywhere. It was out there. But we never saw very high levels anywhere. I think that's very consistent with what Margie showed for the visuals as well. It's just there, it was everywhere, but never got to it. Prorocentrum, on the other hand, we had some relatively high levels by the qPCR. Not sure, not sure what's going on with this, but we did have very high level, not very high, some elevated levels by qPCR. The visuals were not quite as high as what would be in the qPCR, but and they continued right with the Alex and Martin. Okay. And there was some Mycans thrown in here too by visual. Definitely see that. And this is in New York, and a lot of these samples that were elevated for pervocentrum were definitely right during that March Alex, I believe, there in August, September. This is March. You see where March was? You could see that in the satellite images. That is where March was for the most part. I'm sure, you know, there were a few large samples that you guys picked up on other places, but for the most part, the blooms of March were right there. Alex, same thing. We picked up a few samples with Alex in it that we got in here. Oh, by the way, we got 977 samples. But to be fair, you know, we don't look at all of ours visually. We do ours like, right here. So all of, you know, ours are primarily focused here in New York River because we're biased. You know, that's where we get our samples. A lot of the VIMS people. Here are the qPCR results for Pseudonetia. And again, we see Todd showed you much more in depth what was going on for the Pseudonetia by the visual counts. And we've had some concerns, and Gail has been working really, Gail Scott, my lab manager, extraordinaire, by the way. She keeps me straight, she takes care of everything, but she has been working really hard on the qPCR assays for Dinophysis, the Pseudonetia species, and the Azadinium Epidoma group, the acid producers. And she's been, we've been bothered by the Pseudonetia and the lack of correlation between what been seen in the Pseudonetia visuals and what we've been getting in our qPCR assays. And some of that I think may have to do with differences. And so I'll get back to that. This is the Dinophysis qPCR, which actually this assay seems to be pretty robust, and we've seen some more consistency with what you're seeing visually. But of course, those counts are very low. Okay, so qPCR counts are also very low. But that's what you would expect for Dinophysis. Thank goodness. Thank goodness we're not getting high counts. And this is that amphidome. That's what I've been talking about. Anyway, oh, here. So, Pseudonetia species, the visual and qPCRs were not as well correlated as we would like with that Penna et al. 2007. I mean, sometimes they were okay and they were coming out pretty well, but we went ahead and re-blasted. You know, Gail came into my office one day and she just said, I'm just going to re-blast those primers again. We did it, of course, when we first looked at these assays a couple of years ago, but there's a lot more stuff that gets put into GenBank every day. It's a database, it's updated all the time. So she re-blasted those primers, and sure enough, they came back. You know, there's a lot more things that have been put into GenBank, and the primers from Penna et al. 2007. Not a surprise, this happens. Turns out they were not very specific. So we went back and looked at the other assay that we had tested at that time, the Fitzpatrick assay 2010, which when we compared these side by side a couple of years ago, we had decided that the Penna looked like it better, but the Fitzpatrick primers look now like they were more specific. So we said, okay, it might not be as sensitive as what we had first, but let's go back to that because it looks like the primers are better, might be more specific. So let's go back and look at some of these samples from way back when. Okay, now we've got this problem of from the ODU lab, these what I call the skinny and fat Dinophysis, okay? The ones that are smaller, but the fatter ones are the ones that they could be producing, as Todd was talking about, the oxalic acid. I mean, the Demoic acid. This is the diaplasis pseudoinitial. Okay, so for the old assay, it looked like for this, for these guys, it might be better. So what are we gonna do here? I don't know.
So you're saying the old assay was better for the fat ones,
Possibly,
and then the new assay is better for the thin one?
yeah, maybe
That's what you're saying?
yeah, possibly. If you look at this,
It's not funny.
I don't,
You gotta do two.
yeah. That's what I'm wondering, as I'm looking at this, right? Because Gail's saying, oh, well, it looks like this new assay is better, and it does correlate better for some of these. Okay, but look at these. I don't know. Because you've got these kind of counts. Okay. So we have to look at this more closely. I think those of us who are working on your feet is valuable. Right?
Quick question, how do they separate that versus, I mean, is it visual or?
Visually,
Just
when they do the visual. So that's my one.
You measure them.
Yes, we
Yeah.
have a camera that we can measure more precisely. And then we have the little the scale in the
If they're not
eyepiece.
acidic or anything like that.
Right, yeah, because. It might go like
Yeah,
this.
You know, a lot
Anyway,
of the cells are right on the cusp
Hmm.
of one side to the other. So maybe
And the only way
we'll
is
sit down and talk about
doing molecular analysis.
I'm sorry.
The only way to know is to do some molecular analysis.
Yeah.
To separate between.
but I think, anyway, I think it's something we need to. Look at.
Yeah.
And I think maybe we need, these were 2021 samples that we went back and ran. And I think we need to do maybe some correlative analysis with the toxin levels. And correlative analysis with the thin and fat, bones and fat cells, and look at it. That's what I was thinking. But now we have a digital PCR machine. And there are a couple of the assays that I definitely want to run through with the digital PCR machine. Alex is one of those that I want to see on the digital PCR machine. We now have several years of Alex cultures available to look at their using the digital PCR machine. Maybe the student initiate is one of those assays we should also that digital. Maybe large assay with very robust, but maybe the first. But that's just my thoughts on it. These are some things that I presented at the HAB symposium, but I thought I'd just go over it for everybody here. Can everybody hear me on Zoom? Is that okay? So I'm looking at, this is changing directions quite a bit from talking about molecular assays. For those walks of us who want to talk about molecular assays, but this is looking at the impacts. We did some study looking at the impacts on oysters of the late summer blooms of Marge and Alex, Margaret and Alexandrian Monoleta. And Sylvain Gaillard will be presenting this afternoon, who's a postdoc in my lab, some really nice data and some work he's been doing on the effect of Alexandrian Monoleta on oysters and on the gene expression in the toxin GDA, its uptake and distribution in the tissues and on the gene expression in oysters. So I'm just gonna give you some field data from some studies that we did. And this is looking at what happens when these blooms occur, these really dense blooms of marginales occur in the late summer. Because we were working with aquaculturists here in the region and deploying oysters at their sites. And so these different bars are just the different sites where we deployed oysters. We had a reference site, that there's a whole story to that. That was what happened in 2021. Reference site became a non-reference site when the bloom actually occurred there of Alex in 2020. But what happens here is this is the change in shell height that occurs as the oysters are growing. And what happened here is as the bloom set in, the oysters stop growing. Okay, and this usually happens because the oysters shut down. Okay, they sense something that's not good for them to be eating or not good for them to be filtering, and they shut down and stop filtration. And so when that happens, they clearly will stop their growth. And that is what happened here. And my graduate student who was working on this project ran models for it and showed that the growth rate is indeed lower as larger Alex concentrations increase. Okay, when either of those organisms are around, neither of those are around. And the impacts lag the onset of those blooms by about 2 weeks, which is what you would expect, actually. That's an effective result. And the oysters that were deployed, and these samples were run in Juliette Smith's lab. They ran the analyses for the toxin. So thank you, Margaret, for running these analyses. So the toxin Galliodomine. And what we see is those oysters at these sites where they were deployed. Indeed, in 2017, when we had a bloom of Alexandrian that produces this toxin Galliodomine, they indeed, when they started filtering again, because they can only stay shut down for a certain amount of time, they take up this toxin and accumulate it in their tissues. And Sylvain will talk a little bit this afternoon about what happened in the lab. He was doing an experiment with oysters. And in 2020, the same thing happened in the York River here. The oysters took up the toxin, and then over time, they began to get rid of that toxin. But this is even a month later, they still have some residual amount of toxin in their tissues. And the same thing happened at our control site, where there was not supposed to be a bloom, because there hadn't been a bloom there before, but we had oysters deployed at a control site. And indeed, there was a bloom of Alex there in 2020. And we looked at some of those oysters, and indeed they took up the toxin as well. And then a couple of weeks later, and a month later, about 6 weeks later, we could no longer detect toxin in these oysters here. Intertidally, there was still a little bit of residuals. One of the oysters there. So this is just a summary of what happened where 2007 through 2012, we had blooms of marginales in the York River. 2013-15, Alex started to spread throughout the lower bay, and then we've had blooms throughout the bay in 13, 15, 16, 20. No blooms happened in 14 or 19. Blooms of large only in the York in 10 and 11, and in 21. There was a bloom of Marge only in 2018, but only in the lower trips, like in the James River, the Lafayette. We didn't have it in New York. And then this year, we had bloom in New York primarily, of Marge with some dense Alex patches here and there. And then this mixture looked really strange. So 2022 blooms in New York and some wrath were dead, the first mix of species. Spring, early summer, and then this Marge bloom that persisted for almost 2 months in New York. But it was a mixture of Marge, Proro, both Proros, Alex, even some heterocapsa. We found some heterocapsa, rotonata thrown in, even with Marge here and there. It was very weird. New pseudonitidinous assay looks promising, but we may have to do it with the old one, so stay tuned. Oyster growth rate slows after the onset of Marge and Alex blooms, and oysters exposed to blooms of Alex accumulate the toxin, DDA, and deteriorate slowly after the bloom dissipates. And lots of support for all of this work. I mean, DDH in particular for all of the monitoring that we do, HAB and NOAA aquaculture for the studies that we've done. Thank you.
Now we have about 10-ish minutes for Q&A session. That's the end of our marine biotoxin recap for this morning. If anyone has any questions on any of the presentations.
I did want to confirm, I just got an email from one of my students that the bloom did persist in the Lafayette River at least, but it was sort of come and go.
Okay.
As it initiated on the 22nd of July and stayed around from the beginning of September.
Okay, so it's
Yeah,
similar.
but it wasn't real high concentrations and not every time.
Yeah.
It's in close to me.
And it never made it out into Elizabeth either, really. Like,
Not really.
That's a lot, yeah.
Yeah, so just we had reports of it a little bit in the river, but it just sort of hung around, but it wasn't really nasty.
Yeah, and it's mixed. Too, like,
There's a mixed bloom?
Yeah.
I think it was mixed.
Yeah.
And a lot of the shellfish ones were really mixing.
I don't know, maybe, Andrew, you know, like, I don't think there was any fish kill reports this year.
Yeah.
That I was aware of, or,
Well, so not official reports, but I was talking to some locals.
Okay.
And they said this is the biggest one they've ever seen, and that they had buddies who were crab potting, they're full of the crab pot.
No, that's the yours.
in, like in, York proper and Center Creek and, you know. What about bioluminescence? Was there any bioluminescence? I don't know.
And here at and
Okay.
I checked every single night, and sometimes it was really bright down at New York County. And there were a couple nights where it was pretty bright.
Didn't make the news as much this year, though.
I didn't hear anything about the ocean front or.
We were actually on the news, so maybe I can't little video of Thomas. And on Twitter.
One thing we did too in our lab is we did make a QR code for bloom reporting. We haven't deployed it yet. Because, you know, we're just trying to figure out the management and all that, but we were hoping that maybe we get more reports of bioluminescence in particular, or discolored water from the estuaries. If we deployed those QR codes like at marinas and stuff for people on the water, because in the lakes, it's easier, people notice the blooms more, but not so much in.
In years past, when it's made it down and it's been up along the Norfolk beaches and in the ocean, there's definitely, we'll get reports.
You get.
Yeah, we'll get stuff. I think it just wasn't there.
Yeah.
I went to talk to a high school class this summer, and I learned that they did see a bloom in 2021 of Alex. And I think it was and because this kid was telling me he was playing around in the bioluminescence in the backyard, I said, when? He said in 2021 September. I said, are you sure? Do you have evidence? And he showed me a video. I said, can you send this to me, because we don't have it recorded. And so if we put this QR code in.
I think it's a really good idea because we just miss things like that.
Yeah, we
You know,
had it,
no
we just
person
had it.
would know.
Yeah, we just gotta sort of populate it, and we figured if it's coming to us, then it'll be less threatening, if like it's going to BDH and people think, oh no, I'm gonna post it, we'll be able to put this shit in. You know,
Well,
just to report
Bioluminescence, you know.
Yeah. Yeah,
Last thing I did this, I was team up with my significant other who was learning to fight lanes.
yeah.
And so we got an aerial view of all of them, got the level a couple times, and
That was the
actually
photo I had at the beginning.
recently, they sent me a picture of heterocapsid that we were,
Yeah.
didn't know about where it was. So, and the training facility, they said we happen to have scientists. And all the other pictures of
Oh,
the
nice.
training. So that'd be a.
So if you wanna go flying,
Yeah, yeah.
I mean, I don't know, I know it's like at some point, like Shelley commented, like they're looking. Maybe there's some, you know, aim out the window with some of their barometers too.
Well, another thing that we're doing is we're working with NASA Langley, that they wanna fly drones that have support. And so we're gonna try this summer, well, this spring, do some test flights with this team of drone operators there, and then.
Talk to them, yeah, and we never got anywhere.
Well, we had this plan for summer, and then they had to wait till the tower was closed, and then the tower was always open. Every time we got the HRSD boat, we were gonna do data flow underneath it and go out ourselves, and every time we would get a last minute thing, oops, they're flying, can't take off. So now they're trying to think of other places to take off from. So it's not just.
Yeah. We're pretty much.
Is it your code for the movement reading?
Well, I'm just gonna say, well, today we're around.
We have.
But we just. We need to do this.
For central information. Did you read out the center?
On Zoom, okay. I can see everybody who's on.
It's, you know, he's been trying last night, it's like, he seals on part of it. I just wonder what he wants it all.
Yeah.
So just as a reminder that all of the presentations and the recordings from today will be made available sometime next week on VDH's website, so full information will be available.
Yes.
Everyone wants to get more tea or coffee and then there's no more questions.
Yeah. Like in terms of some.
Yeah, we'll play.
We don't usually do that. So that's.
Yeah.
Yeah.
We use that sort of thing. Then Scott, I had a bigger.
Yeah.
I don't know, they
Yeah.
use slumbers.
It was a-
Like,
yeah.
that's- We haven't gone.
You should say. Like the rigor. There's like they're taking some products as well as do that.
We do have a here. We just had a graph in that here. Also classified it, right? Yeah, that would be. So for the most part of the deeper water. They're not. I'll be there now. That's why we're fishing. But not that that is not. It's for the microscopy. If it's something that's gonna go.
Yes.
Toxin one, we got it produced that. Something.
Yeah.
It's just that,
I'm not worried about the I think that's the very good point, but like, it's crazy that like sneak activations and stuff like that, like. After through that.
yeah.
You know.
Well, we have a bunch of sales that are lower than they have, like, a little more. There's onward.
We're like.
Still.
Yeah.
A lot of places that to go there.
Yeah, it's just running, it's like Vegas, not an.
Yeah.
We don't have, it's obviously.
Something that.
Yeah, that was. Bigger though. I am.
Andrews is. Thank you. Yes.
That's where the issue. I call it. she's presenting like a long. I think it's her state level. I think folks were going to understand. And then just one of those water. Even I was sort of scared. I said one was like, well, it's pretty young, but even at the level, say, they follow that. Then if you start with the quality, you get the concentration around. Yeah. Okay, yeah.
Next, we'll be starting the freshwater biotoxin recap section. And first we'll have Leah Anne Gibala-Smith. She is with the Mulholland Lab freshwater recap. Leah is an aquatic ecologist and algal taxonomist who has over 25 years experience related to water quality issues, drivers of aquatic community structure, and identification of phytoplankton and algae in fresh, tidal, and marine systems. He ran a water quality and nutrient laboratory for over 5 years prior to joining the Phytoplankton Analysis Laboratory at ODU in 2014. Since becoming the laboratory supervisor in 2016, Leah has worked with the Virginia HAB task force in conducting taxonomic enumerations and toxin assays in the state's fresh and coastal waters for both monitoring and rapid response efforts.
Thank you. So we're changing gears a little bit with moving from our coastal and tidal areas into our freshwater system in the state. Just gonna orient myself here. Use the keyboard, yes. And I'm with the Phytoplankton Analysis Lab at ODU in Norfolk, Virginia. Margie Mulholland, Dr. Mulholland is our PI, and I'm her supervisor. You see a picture here of Catherine, who you saw give the presentation last year. She's up in Canada with all her snow and wind, and I returned back to the lab to give you the presentation today. And we have a whole new set of undergrad interns that are helping us do our samples that Margie mentioned, and many of them have research projects related to some of the imaging technologies that Margie was talking about earlier. The Phytoplankton Analysis Laboratory has been involved with the harmful algal bloom task force for about 30 years. We work very closely with our friends at the Virginia Institute of Marine Science, as well as the Department of Health. And as we all know, algae blooms are kind of hard to find. They're hard to, when do we decide what is what? And that's some of the things that we're working on together with my cell counts and everybody else's qPCR molecular methods. Harmful algal blooms can be considered harmful for a lot of different reasons, but what we're primarily concerned with in our lab are the production of toxins associated with human health illness. And one of the neat things about Virginia is we have a continuum of a salinity continuum. We have the coastal, we have the tidal, and we have the freshwater. So I really get to spend a lot of time with a wide, diverse array of different types of phytoplankton species, and I love it. It is so fun. And we are concentrating on my talk today on the freshwater HAB events that are mostly cyanobacteria, but every so often we'll have a HAB event where samples come in and they are not toxin producing cyanobacteria. But like at Smith Mountain Lake, they had a lot of euglena this year, and we've seen other types of blooms in the state over the years. These are our main potentially toxigenic cyanobacteria that we do see regularly every year in the state. I actually forgot to label the top two, and I'm wondering if our friends over here maybe stay down and tell us what those are.
The first one's gonna be Microcera.
Yeah. And this one, microcolia, the Shenandoah. So that's kind of something that's new, and everybody's gonna be exploring some of those benefits in the future. One of the things we've been doing in the lab and with our partners is at VDH is kind of thinking about, in addition to taxonomy, what sort of toxins these groups are associated with, and so we've been keeping track of that. And you'll see over here on the my move it over to well, the bold ones are the ones that we see on Lake Anna, because everybody likes to know about the Lake Annas. So every time I'm always thinking about, oh, did we see that on Lake Anna? So, oh, here we are. I'll probably lose it. These are our major, oh, on here. I'm not good at this. Oh my gosh. All right. I'm just gonna go on. So what we do in the lab for our freshwater HABs, what we do this for a lot of our other samples that come in, is first we do a quick scan, we identify what we see, what are the dominants, is it really a cyanobacteria? Is it a toxin producer? And then we focus in, we do individual cell counts to get VDH of their cell counts so that they can take action if they want to. And then we also run toxin assays. And something that we're doing now is we do all four toxins for each sample that comes in. And you see this picture, that's the new CAS cube that we use with VDH. And that's one of our undergrad interns, Marcus, helping me do a calibration. And this has been really, really helpful with managing our workflow during the HAB season in the summers, because it's a multiplex system. It can do up to 3 different toxin assays at a time. And we still have our manual reader. And so what's really nice is you can toss the 3 long ones on there and then do sacks of toxin by hand. And then you can potentially do all your toxins in one day. And that's really good for workflow in the summer when we have a lot of students that are out doing research or things that they do. One of the things I wanted to show is since 2013, we've seen a steady increase in the amount of analyses we do. And so I'm not talking about samples, I'm just talking about analyses. So that includes taxonomic enumerations, microcystins analysis, cylindrospermopsin analysis, Anatoxin A analysis, and saxitoxin analysis. So again, this is not number of samples, this is number of analyses when we're thinking about workflow. And so back in 2013, we had a handful, and 2019, 2020, it really ramped up a lot. The last couple of years, it's kind of slowed down again. And I'm not sure if that's just people. I think sometimes maybe a lot of times the public thinks, oh, well, that was reported, they're seeing, they come here all the time and look at it. So sometimes I'm not sure if that's an artifact.
Yeah, that's us listening to you and saying, we're trying to reduce the
workflow. So in 2022, our lab received 549 samples under the contract, and 65 of them were freshwater. We conducted 313 analyses on those freshwater samples. We did taxonomic enumerations in all 4 of the toxins. And how about this? Sorry, guys. I think there's a slide missing, but
Take your time, you have plenty.
yeah, I have a printout of it. Oh my God. Okay, so we'll hop over to Lake Anna for a minute. What I did this year, because we sort of had a low event year, I thought it would be fun to revisit Lake Anna. It's on everybody's radar right now. And so I'm just gonna review our taxonomy at each of our regularly sampled sites since 2017. There's been about 23 different sampling locations over the years, but only a few of them are visited every year multiple times. And those are the ones that I'm going to discuss today. You'll see on this map, there's some on the south side. Those only were visited a few times. And I think DEQ is going to talk more about the sampling effort later. So once again, I'm only going to talk about these 7 locations that we see every month on a regular basis. This is a nice map that I've borrowed from DEQ, and they divide the lake into 3 main sections. And so our lower section of the North Anna branch has 2 locations, and over the years, and you'll see for all the sites, the highest peak, the peak of maximum total cell counts occurred in 2019 for all locations on the lake. And here in the lower section, the North Anna branch, we see a lot of Rhaphidiopsis and Prentolambia, but no major neurotoxins are usually detected on. And when we do find any toxins, they're barely above the lower threshold of the tests. But we very rarely see any of the neurotoxins here. On the western section of the North Anna branch, it has a much more complex cyanobacteria community, and we do see more positive hits with anatoxin A. And I think that must have a lot to do with the species that we see there. And so here in the western section of the North Anna branch, once again, you can see on this part of the lake, the highest cell count densities were observed in 2019. And it's mostly dominated by Rhaphidiopsis, like the rest of the lake, but we do see a really mixed community here. And some of those are known neurotoxin producers. And that includes Pectenotoxins and Crassostrea, as well as a whole number of different types of Phylloxera. And this section of the lake, I have observed Spermopsis a few times, not every year, but on years that we do see it, there seems to be a higher nitrogen fixing component in the overall community. And it's really cool. And so again, 2019, you see this huge spike, and in 2022, Catherine, and 2021, Catherine had quite a few neurotoxin hits on her sample. And you'll see that big green, bars, and that's associated with that Pectenotoxins, which used to be a fan line. You guys look at all of this. And then once again, we have a very diverse section of the lake at this sample as well. But our highest cell counts were observed this past year when she was running the lab. And so I thought that was interesting that we had that spike, not this at this part of the lake, higher up on the western branch. The 2019 spike was not as high as elsewhere on the lake. And now we're up in the northernmost section. When I've done a tour up there seems to be a lot more agricultural things and larger properties and larger and big lawns and things like that up there. And cows and all kinds of things. And so here, we did still see a few positive hits for Anatoxin A, but not as many as we did on the western side. And we had a little bit, and again, everything was dominated by Rhophidiopsis, Pseudoanabata, but we do see the Cuspidothrix up on this part of the lake as well. Now, something that I had not seen since the first year or two that we were looking at Lake Anna, Catherine was finding this past summer, she found microcystic, very, very low component of the community at large. Like we're talking like 400 cells of microcystic to like tens or hundreds of thousands of the filaments. But I thought that was interesting because I had never observed microcystics on that part of the lake previously. So we'll have to keep a really good eye on that for this upcoming season and see if we see that cropping up. And as you get further up the branch, where it's narrower and shallower and warmer, you see this really much more diverse community. And then that's when you start seeing more variability in the toxins that you get a positive hit for above the lower detection limit of the test. And just because, and just remember, these are really, really low toxin levels, just above the detection limit of it. So again, we had the big cell count spike back in 2019, and we had nothing close to those cell numbers for the last few years. But we again have a really diverse community, and we're starting to see higher up where it's even more shallow, even a warmer water. We're seeing more of that positive Anatoxin A hit on the assay, and again a lot of species diversity. But you know, Rhophidiopsis is still there, Pseudoanabata is still there, their dominance. And then this one section here, 39, is a little bit, it's around the corner further north. And then we see a little bit less species diversity in here, but we're still getting a little bit of Anatoxin. And in general, these are my general thoughts about the community on Lake Anna, is that the toxins almost always are just below or just at or above the reportable detection limits when we do see it. And we're reminded that microcystins is not a good sentinel for the neurotoxin. They don't always occur at the same time. And it's just a really diverse system, and it's really, really, look at these pretty things that we have here too, because they all look very similar to each other, but they do different things and they behave different ways. And so this Dolichospermum, the Sphaerum spermopsis, and the Anabaenomopsis, they're not there at the same time every year. So understanding Lake Anna phytoplankton community is very complicated. We are looking to incorporate some student work. We have a Leah with us in the audience today. She's Margie's new PhD candidate, and she's been doing a lot of work isolating cyanobacterial cultures to examine growth kinetics, and that might speak to some of the toxin production that we're seeing, that might speak to some of the community structure. We're also talking about with some of the folks that are working up there already, thinking about seed banks that might be feeding into these regular Dolichospermum dominant blooms in some of the back coves, possibly developing some qPCR methods, and something that I'm really looking forward to in terms of like an outreach part of our relationship with everybody in the lab is doing a QA/QC for taxonomic nomenclature with the LACA group. They're starting a volunteer group and they've invited the HAB coordinator from NOAA to come up, and I'm gonna work through taxonomy with them to make sure everybody's calling everything the same, because depending on who trains you, there's different names. And let's see. Now, I don't know why, I'm missing a slide, so I'm just gonna put this picture up and grab the printout on my.
We'll save it on the email.
Let me save it over. Sorry, guys. So, sorry, guys, I must have saved an earlier draft, but we had some other lakes that had algae blooms besides Lake Anna. We had Nottoway Lake, which was dominated by Virginica and Crassostrea, and that had low toxins. They all had low toxins. Smith Mountain Lake was dominated by Sudanabana for the cyanobacteria component, but there was Catherine observed a lot of euglena in those samples. Lake Gaston, which we do see periodically with filamentous cyanos. Lake Gaston was dominated by Planktolympia and Sudanabana. And then we had Lake Ashby, which was one I had never seen when I was here previously. And Catherine identified Planktolympia and Rapidiosis on there. And again, there were low toxins associated with the area. I'm sorry. Here we go.
Thank you.
You have some time if anyone has questions for Leah.
Oh. Yeah, again, just gonna grab some water.
All right, up next, we'll have Margaret presenting VDH's freshwater advisory and alert recap for 2022. Margaret has served as the Waterborne Hazards Program Coordinator at VDH for 7 years, overseeing the coastal beach monitoring program and helping coordinate members of the Virginia Harmful Algal Bloom Task Force and supporting outreach and education for waterborne recreational water illness prevention.
We'll get the next time. So, a little overview of what we're going to talk about. You're going to hear more about Lake Anna and the river, of course. We're going to do a recap of all of our advisories, as well as some of the new management strategies we gave you this year. We're going to highlight some of our meetings and other efforts that we help coordinate to bring everyone together, get all this stuff done, and talk a little bit about kind of what we have prioritized for the upcoming year. There's a lot going on here, and obviously I don't expect you to see this. But what we have here is a little bit, the flow chart is from our new algal mat alert job aid I'm just gonna call it algal mat alert strategy that we came up with this year to help us better and communicate with the public regarding risks involved with algal mats and bacteria. Obviously, when we met last year this time, I think it's February 25th, we met last year, virtually. I talked a lot about the Northwest Shenandoah River. We'll talk more about it today. I can't get away from it. We had a pretty large scale advisory issued in 2021. And this was in part due to a lot of flexibility that we had just put together in our cyanobacteria advisory guidance in 2021. So we had just hot off the presses, had a lot of flexibility in terms of how we were going to manage algal mats as far as public health is concerned. But it ended up being a little too flexible, and what resulted was a very large advisory to be to err on the side of caution. One of the things that is that led to that long advisory, 52-mile advisory in the North Fork was because we had several drinking water intakes we were concerned about. We had several public health complaints and occupational complaints of staff. And we also, just as we had developed a method for analyzing mapped samples for toxin, we had a few samples that we were able to collect and analyze, but then due to some health concerns, we had to stop that analysis, and we didn't have the capacity to seek that support from another third party lab. So we sort of decided we need to err on the side of caution, and so that's what happens when sometimes you're a little too flexible with guidance, that you end up being a little overprotective, possibly. That could be an interpretation that was an overprotective advisory. But at any rate, we did that extent that we chose from Chapman Boat Landing to Riverton Boat Landing because they were landmarks, and this is a fairly rural river where there's not a whole lot that we can say, hey, this point to this point, rather than just saying lat long, we tried to pick focal point people would do based on the boat. So I wanted to kind of intro that a large part of 2021, 2022 has been developing this algal mat strategy, which requires just a massive amount of adaptation on our part. On the part of our colleagues who we work with, huge learning experience. And we are so grateful to everyone that's joining us, helped us throughout this process, because this was a lot. And we're very grateful that we have now arrived at a place where we feel like maybe this is a good strategy to stick with. And that guidance document that we had just issued in 2021 for the advisories. So we have alerts and advisories now, two different strategies. They are going to coexist together. Right now, they are separate documents, and we're going to work on coalescing them. But when we met last year, we were trying to figure out the whole algal mat alert strategy. We really didn't know what it was going to look like. And so we're going to talk about it here today, that essentially we still have our cyanobacteria guidance, and that still works and plays into the new strategy for algal mat alert. One of the things that we needed the algal mat alert strategy to do was to kind of clarify for the public what we felt the immediate risks were, while sort of tiptoeing across that balance of not restricting all the use of the river. And so the way that we sort of did that was the algal mat alert strategy breaks out a different category from advisories, where advisories were saying you don't recommend swimming. With alerts, we're saying you may continue to use the water, you may continue to swim, providing you avoid contact with the mats. We also have a caveat about that we don't recommend that you take your littles or your dogs to this water body. And so as part of developing that strategy, we have some signage that you can kind of see. We've got some large signs that we can place at water access points, as well as handouts that are a little bit more picture oriented and helps kind of get across the language barrier. That sort of details what you should and shouldn't do in that book. The other thing that we needed to do, aside from communicating the risk, is that we needed to make it clear we didn't have the capacity to collect or analyze the MAC sample. And so this strategy involves only collection of water samples. And we are still collecting them as we investigate those HAB MAC complaints. So we also had so we're not going to collect and analyze MAC samples. Then to make sure that we're protecting the public, though we don't necessarily have to have toxins presence, if we know how to identify a cyanobacteria MAC, that in prior years we had some analysis show us that we had some high toxin concentrations, we can use the presence of it as a rationale to issue the alert alone. Now with that, it's, obviously we can have a lot of patchiness in these MAC blooms, if you will, across the river. So the language in this new strategy details that when DQ goes out and they do an investigation, it's not just patchiness that would get an alert. It would be widespread and unavoidable patches of these MACs. So if you can't walk into it and avoid it, we shouldn't be recommending swimming. And so that was another caveat that we included in that strategy. And let's see. The flow chart was particularly helpful when we started reaching out all of our groups to talk about it, to get feedback. This included the Shenandoah Riverkeeper, Fairfax Health Department. We met with them several times since they were affected by these MACs in particular. We talked to our Office of Drinking Water, obviously, about it. And we didn't get a ton of feedback from any of the groups that we talked to. And so since we seem to be working and moving right along with this, we're planning to proceed to sort of coalesce the algae alert strategy with our existing guidance with some minor changes along the way. So felt like I needed to kind of introduce you guys to this new strategy. You probably folks have already heard this and I'm tired of hearing about the North Fork and strategy for that. But this kind of sets the stage for the next slide. So again, a lot going on in this one slide. What we have here is a map provided by Todd. Todd is the map man today. The map just displays where we had our alerts and advisories issued by the this year. The red is going to correspond to advisories and the yellow is going to correspond to alerts. And as Leah has already kind of gone through these water bodies already, we have an alert for the North Fork and Nour River. That color dot you see in pink is going to correspond to the row with a label on the bottom for that water body. As you then look at the rest of the table at the bottom, you'll see that we have the month samples. That kind of shows you across the season, how season we define for recreational use as May through October. The X's indicate sample that occurred in that particular month. And then the red is the red bars in the graph are gonna align with when we had advisory issues or ongoing. And the yellow for the North Fork at the very bottom is when we had the alert. So with that, the final column in that graph is the total number of days under advisory or alert. And one thing I want to point out is that Lake Anna, we have several dots, that's because we have several polygons or water body sections we break Lake Anna up into. So we try to, and there's different communities as Leah had mentioned, so we try to separate them out. But I'm just considering that one water body. There were differences in terms of when we had advisories for Lake Anna, so that allows you to kind of parse out when the upper sections of Lake Anna tend to go under advisory first and get off last because and be more concentrated. The further up the lake you go, and then the lower segment of as you get closer to Route 208 on the Lake Anna, you would have smaller durations of advisories. Okay. These sampling events here in this gray box, it's days that water body was sampled. So there was a sample event on XYZ date that counts with one. And I'm not counting individual samples, but this is just to kind of show overall workload in terms of the different water bodies. Chris Green is not a HAB task force water body that we didn't provide the sampling at that location this year. That is a water body that is in Albemarle County. The 62-acre lake and managed by the Parks and Rec system. So they have a contract with a private management firm that conducts the analyses and lets us know the results to help them make the decisions and work with the local. Let's see. So we had total of 3 advisories in the works, and these were either due to the presence of algal mats that were widespread and unavoidable for the North Fork, and then the red water bodies would be because we had detox tax up over 100,000. We had 0 samples, and that's a big deal. Last year we did. I'm glad to say we had 0 this year. As Leah said, basically all of our samples are coming back very low in toxins. But we do have high biomass percentages. And so that's why most of our advisories are due to the cell counts. We have a hybrid system for issuing that involves cell count and with Leah Lake, the only thing that I'm going to mention is it's 188-acre impoundment, and it's just north of Nottoway, Blackstone, Virginia. It's managed by the Department of Wildlife Resources, and it's not used for swimming, but there is a boat ramp there. So there's a very light human exposure that is supposedly occurring. And as you can see, it's one of the water bodies that we only have one sample for. One sample event. And it was because we are in a situation where we have to prioritize our resources after working through with the local health department that it's appropriate to issue this advisory. It meets our criteria for issuing an advisory over that PFOX threshold, but it's not high rec use. DWR felt that they could manage it with some signage. We had already issued the advisory. So we decided that in order to protect our resources, to let the advisory stay in place until the end of the season. At the end of our season in October, we lift all advisories as of October 31st. And so that's kind of when we end our response efforts. And that's when we would sort of get our count from going back in time through the calendar to get these numbers that you see. So we're not going to leave the advisories in place because we're not plugging samples. So Lead Lake is important because between that and some other events we've had over the last couple of years with BCR, we are needing to better prioritize where we provide our services. And so we're not only going to be looking at is the public water body, but is there also high recreation use there? Because if it's just used for fishing, we can deprioritize that if the resources don't provide, are not there for us to go out. In short, so that's also kind of come into play as we are updating this guidance document, is that we're gonna refine our response criteria, so to say, as it relates to refuse. So now we're gonna just highlight the events at North Fork and then of course Lake Anne. For those advisories that occurred there. On the North Fork, we already mentioned in 2021, we had a large advisory. In 2022, we're now using this new management strategy. And we had our first alert issued for the approximate 5-mile extent, and this was around the Deer Rapids boat ramp southwest of Strohm. And this was in late July. Again, the default is 2.5 miles below and above that site. So that gives us a margin of safety in terms of our workload. What we were seeing in 2021 is we would go out and we would sample and we would have hotspot. And then next week we would get another complaint a mile away. We have to go back out to that hotspot, and it just kind of kept growing every week, and it's like constant HAB complaints coming in, and we just kind of decided we need a little bit more space in terms of our extent for the alerts to make sure we're not, our workload can be there. So that initial alert went out for 5 miles. And then a couple of days later, it was within about a week, we had gotten additional reports that we had some mats present below Strasbourg and above Strasbourg. And so the investigation also included the town park at Strasbourg. And the mats were widespread and unavoidable above and below Strasbourg, but they were avoidable at the town park. So that's why we have a green dot for the town park, whereas the other sites have the yellow triangles for needing an alpha alert. Widespread unavoidable mats that are visually confirmed to be biotoxin. So this is a larger version of our alpha alert signage that was posted at all of these sites, even though the town park did not have widespread and unavoidable. It fell within the area between 2.5 miles above and below each site. We had one contiguous area as a result. So we were able to sign that town park site. So we did coordinate with Office of Drinking Water so that they could work with their field office and their public utility for the intake. It's not very close to this site, but we didn't hear anything in terms of concerns about the levels, and any time we issued an update, a status report and posted on our website about this alert, we checked in with the queue to make sure that there had changes or updates. And they had stated that there were no scrutiny or as a result of this alert. So this is just a comparison. This is a comparison map to kind of show you the 2021 advisory and the 2022 alert. In the 2021 advisory, there was a hotspot at what we call the Bethel Road area, just kind of in here. I know you guys in Zoomland can see that. Okay. Is it showing? No, it's not. It's on
You have
here.
to keep, you have to kind of move it around if you find the screen that's on. Yes, good job.
Okay. The Bethel Road areas are roughly right in here someplace, and then these little continuous parts of the river. So that essentially is where, not far from the Deer Rapids, the first HAB complaint we had gotten in 2022. So, finding additional sites below that, also kind of aligned with one, this past year. So the reason I have this range up here for the toxic concentrations is because we first of all, we don't want to compare our mass sample results from 2021 with our recreational advisory threshold. That would not be appropriate. But we show these just to remind everyone. Lost it. I don't know how I lost it. There it is. Yeah. Just to remind everyone that this is why this water body has gotten so much. Was those first mass samples we were able to get. And so when we look at the water samples next to it, those results for 2021 and 2022, they're what we would expect to see. Much lower, obviously, because the toxins tend to retain in the cell wall, the dense cell walls of these meth species, not forming species. So they certainly can be. We don't know when they are released. I mean, obviously, that the meth dies, those cell walls could release and release to the surrounding water. But that's what we need to investigate. We kind of need to know when would that be happening, and what tools can we use. For example, working with colleagues to get those out there, maybe working with the drinking water and their intakes is a great spot to place those, for example, or other locations, maybe at Town Park where we have seen that prior, places where we could place these methods of looking at sort of long-term release versus our individual samples, which isn't going to show us information over time. So this is just something we want to kind of remind everyone. Yes, of course, the water sample results are low, as we would expect them to be, but they may not always be low, and there could be release at different times. And then for Lake Anna, we did not have, we had a sort of a routine kind of here at Lake Anna. It seemed to be very affected by weather and storm events. I do want to just give a brief shout out to Rappahannock Health District. They are the folks that collect at the beach site at Lake Anna State Park, and they work with DQ. Another shout out to them to get those samples shipped off to our ODU lab. This has been an effort since 2018, where we've done some additional monitoring that DQ has done on a monthly basis during the season. And we will talk more about what's going on with DQ at Lake Anna after lunch. So for the advisories this year, we had advisories that were issued first in the upper Monkey and upper Montana. And then I would say that the worst extent, which is shown here from our status report for an advisory update that we sent out on 8/8, it went all the way down to just above Route 208. And that's about as bad as we've ever seen it. We've never seen 208 have a need for an advisory. That's kind of where the upper separated by the lower part of the lake. We did have, that this is also when we had our issuance of an advisory at the state park. And this is a highly used beach, huge population that uses it every summer. But luckily, that particular advisory, this worst case, we were able to lift most of these back to the middle the split, the area that we call the split.
You're over there on the.
So this split area and this area and this area, we were able to live within about 38 days. So that was a pretty short-lived advisory, although it was for the entire month of August. So that state park beach was not able to be used by patrons for an entire month and a really hot month of the year. That was unfortunate. We did not have any state park beach advisories in 2020 or 2021. So this one has just come back this year. And the worst case, this is the worst case extent for advisories. But we ended up in October, when we lift all of the advisories to the end of the sampling season, we still had the upper and middle Morgana and Monkey branches in place. Those were still active due to the sampling that we had in October, but then we lifted them because we aren't going to end up getting more sampling in October. The other activities that we work on during the year, honestly, 2022 was mostly more for Shenandoah River type issues with algal mats and that strategy development. We met several times with the stakeholders to get that through, and we were able to adopt it right before the season got going in June 2022. To coordinate with everyone, that helps make all of this effort possible. We meet monthly with our primary stakeholder folks, and obviously we planned this meeting last year, which takes a few months to kind of put together. I'm not going to say much about this project because it's not finalized yet, and we don't necessarily have the full result, but because we could not take mat samples and analyze them, we were hoping that we would find some kind of tool that would at least say toxin is present or absent at that time. And because we lost our lab capacity for mat samples, we would love to just keep whatever that is of the lab. And so field test strips came from us. We reached out to Urban's. They've now changed the gold standard diagnostic for their name. But between them, ICPRB staff and the Lord Fairfax Health District folks, we put together a scope of work for collecting mats, getting them shipped by the district health department, and then having those analyzed by the company to determine whether their field test strip could be used for kind of little bits of mixing. And so we hope to be able to share more about that soon. I don't have a full summary of that available yet, but we're still very hopeful that that might be something that could be used not only by us for North Shenandoah River, but by others who have map issues elsewhere. And then finally, a lot of the graphics and visuals you saw in this presentation were on a poster that we presented at the U.S. HAB Symposium in New York. Some of the other projects we had, and our priority for the next month or two is going to be coalescing the algal mat strategy with the existing cyanobacteria guidance. I've already kind of mentioned how we're going to make further tweaks on that. Continued collaboration includes working with some of the partners to see if we could have a SPATT project in the North Shenandoah River in particular. And we have an ongoing effort to develop memorandum of understanding between DEQ and DH for the coordination of all this effort. We're the two co-leads on the HAB task force, DH and DEQ, but there's no formal agreement that says we will work together and how we work together. So we're still working towards that. And I started on a guidance for private water body managers to follow in kind of using our guidance that they so choose to issue their own advisory. I hope to finish that off this year. It's just been kind of waiting on me stuff to get that on the website this year. That may be useful for folks. And so that wraps up my presentation. Thank you all for your help and support during hot season and coming up. Look forward to working.
We still have a few minutes if anyone has any questions from either the freshwater or marine biotoxin session this morning.
Yes, Leah, I'm kind of interested in, I think your strict coordination with Abraxas, or gold standard, seems really, really interesting. But I was curious, did they also do taxonomy? And if so, was the mat dominated by Pectenotoxins again, or did you see Microcoleus this year? There was no taxonomy. We do have some very, very, very preliminary summary information that we can share. That kind of details the use of ELISA in comparison to Toxins. But we don't have numbers. No, I was just, no numbers, but I was just curious if you had seen if it was back to the Pectenotoxins again.
I don't think we know what it was, but we did, well, Mike, you can talk more about it, but there was a concern about the matrix too. They also wanted to work just with mats kind of as a material.
Yes.
So that they could spike those mats in their lab with toxins. And so there was collections of visually observed green algae mats and then visually observed cyanobacteria fences that in the same vicinity that were then set up. And so those were going to be tested by them. But all of this was a sort of a volunteer non-funded project. So
very cool.
Margaret, I love the sign that you had up there, for the album at that one. And this goes back to the conversations I've had with Jill up in Kilmarnock, is that's awesome that it's not only what you can't do, but what you can do. And using very good guidance, in a positive light. That was really, really neat.
And Todd has found a sign that was similar to this from another state. Do you remember which state that was?
Probably California.
We just kind of.
But then but we worked with, was that our graphics department that helped?
Our graphics department kind of took what we envisioned and made it fit for all the uses that we wanted to. Yeah. We had hoped to have even less words, like small words on the bottom. But we couldn't get around those. I like things that are kind of universal imagery.
But just the other thing, when we're talking about focusing on these water bodies where swimming is the major concern, we were getting into a situation with some of these that, we have no enforcement. We're putting out advisories about no swimming. Some of them are managed locally where swimming's prohibited. So this seemed to be counterintuitive that we would be testing the samples to then put out a no swimming advisory in waters where they were being managed for no swimming. So that's why I think by having some of these more general concern signs, if there's a concern, you can just jump right to that versus going for all these hoops where there's no swimming, allowed in the first place.
There's a question online that could be for Leah or Margaret, but Drew wants to know if there are any conjectures as to why we see so many high cell counts of cyanos in Virginia, but almost never any appreciable toxin concentrations.
That's actually a great question because we don't know.
Yeah.
And I think it would be good, actually, one of the reasons Leah wanted to come is, we would love to get some of these in culture and look at controls on toxin production and see what those are.
The one thing I would say, like in Lake Anna, perhaps, is that, those stations are almost always gonna be in the middle of the lake. So that there's a lot of times with cyanobacteria blooms, you can have accumulation along the shore. So it could be that there are denser blooms out there and scums that, maybe aren't being monitored with this, but again, we don't know.
And then also maybe thinking about cell counts of what species, like if you have this high cell counts with
the three that I showed you, the Anabaena-opsis, Sperma-opsis, and the Phylloxera, those high cell counts would be more likely to be associated with some toxin positive results, whereas it seems like the Pseudoanabaena and the Rhaphidiopsis, we wouldn't normally maybe see positive hits. So it's not just cell count alone, but what's getting us the cell count.
Yeah, and I mean, I think that's an ongoing discussion we need to have about our defense list and our hybrid criteria.
It's so hard.
And there's some conjecture too that toxin production, it can be intracellular or extracellular. And you alluded to when it leaks out of the cell. But, you know, maybe at different parts of the bloom cycle that their cells are more leaky or less leaky. You know, compared to, you know, when there's a healthy population, they might keep their toxin in. And then once they're dying or disrupted by something, they might let it all spew.
And then some of the literature I've been reviewing when, you know, you can't sleep at 2 a.m. is the ratio between nutrients, not just elevated nutrients themselves, but just the ratio of the nutrients sometimes can seem to cause more secondary metabolites being produced, be that they're working harder to do this or to do that, and then feel some toxin influences.
There's a comment here by Paul, who said physiological state can alter toxin production by 100-fold. So numbers mean very little.
One thing I forgot to mention, was that I did mention about 2021 in Norfolk when, we had the large advisory that had to do with, some of the health complaints that we had. In 2022, we had 0 calls. So not sure why that would be, but that's just something to let you guys know. We won't have anything to report to CDC on OHABs. We will have, of course, the alert that was issued that will be specific to environmental ones, but we're always happy to not have to report any illnesses.
Yeah. Oh, yeah, Rob asked to please share the BDH sign, the one that says be aware of algal mats. So he grabbed it and drew the presentation with it. It's not very good.
We do have them on our website, under our resources tab. I can throw that up on the meeting website for today. I think we've already said that all the presentations, the recording, all that good stuff, PDFs of the presentation will be on the website and I sent that link already, I think, to everybody, but I'll send it again once everything is posted. And I can throw on that page a link to the resource for all of our signage and stuff. You're welcome to borrow anything that's there. We hope it's helpful. Most of what we have is also borrowed, so very carefully. One minute.
Okay, so lunch should be set up. If you go out this door to the left and go through the double doors, there's 2 sets of double doors. You're gonna go through one set of double doors at the end of this hall. You're gonna make a left, there's another set of double doors. And you're gonna make another left and go into another room. It's called the galley. And there should be lunch set up in there. There are drinks back here in these coolers. There's also water, of course. Not sure how good the coffee is at this point, but you're welcome to that too. And there should be there are tables in there. You can eat in there, or if you want to bring it back in here, you bring it back in here and eat. Yes, there are round tables in the galley room up there to eat. And we should be back here at 1, just start again promptly at 1. Yesterday would have been beautiful outside, but probably a little cool. You can also eat, there's a big table outside. I'm not sure if anybody got this cool. It might be a little cool. So thank you to everybody this morning.
Yeah.
Small things you learn.
Yeah. I should have known, but-
And I think it's awesome that you got dedicated space for futures. So we have a dedicated room where it's potentially file. You can really separate all of our.
Yeah.
all of So we're getting more and more into running videos and running all this. So the idea is we get direction a lot of. And so we were able to build that with respect for the building. We made a huge design flaw that we made incubator room and made it some incubators. We had like double size walls, triple insulation, like for the whole thing. And then we closed doors and it's really hot. And it's still, I know the incubators are stuck. Like, oops. Oopsies.
Oh, my goodness, that's.
Yeah. When you first met me, like I was in the field most of the time, I'm like, never had.
Yeah. It's so different, isn't it? Yeah. Oh, yeah, I remember this thing, it's been like months at sea. Not doing that. In my private life.
Now I go like, so once a month.
Yeah. I did, we did do work where we went to sea, maybe not for months at a time.
Yeah.
Maybe weeks at a time. I was lamenting how, yeah, they used to go out to sea for months at a time. And and then not so much going on.
Yeah, so we need to go monitoring and also no one does that. I mean, it's really.
Well, I mean, I thought it was a few years ago, several years ago now, but when they were talking about the possibility of combining,
You were ready, yeah.
what they're doing, like micro, it's gonna be a PCR onboard platform, integrating that with. And like, that whole, like, no, or it out.
Yeah.
If they were doing that on fixed platform. It was like, it was good, it really was neat, they were talking about that kind of, like, the range like. That every so frequently, they had like 100% of like. Yeah. Yeah.
Yeah.
I thought, oh my God.
Okay.
It's like, yeah. Like, send that off, because.
Yeah.
That'd be.
That'd be very good.
And it will.
Every time I turn around, like I get one little bit fixed and then it's like something else. To be kidding me. It's. Unfortunately. Yeah. Yeah, this year, but we also did it in the water and some. It's 25. Get up ready.
Okay. Oh, I'm gonna direct it. I forgot what
Just.
It's because we're already.
I reminded Dad.
Yeah, can you hear me okay?
Yeah, speak a little more. A little more. Say something again.
Little is it, you say it's a little low?
Yeah, can you turn up your volume? I don't know, maybe we can turn it up on this side.
Yeah, I don't think I have any more powers on my end.
At least we- Okay, can you start sharing your screen through and see if you can see it here?
I certainly can.
Thank you.
Let's see. All right.
See if that works.
Do you see my screen now?
Yes, I do. Let's see,
Okay.
I see. Yeah,
It's showing on the left.
Yeah,
So I'm wondering if it's.
on the wrong display. How do I do it? It's over. Right.
So.
I had that.
Yeah. Okay, I'm gonna let you drive. Weird.
Right. Yes. Right. So it was actually published on. In 2020 or 2022. But on the paper, it didn't come out until January of 2023.
And
Welcome back, everyone. Thank you for your patience. We have a couple of people do our best. Up first, we have Drew Gary with EQ. Drew has served with EQ Central Office Monitoring Team since 2018. And from 2016 to 2018, he served as the DEQ Chesapeake Bay Tributary Monitoring Coordinator. In both roles, he's coordinated and led much of the protocol development, planning, and execution of the agency's statewide monitoring programs. He has served as a member of the HAB Task Force since 2016 and helps coordinate DEQ's role as the primary monitoring agency for investigating harmful algal blooms. Thank you, Drew.
thank you very much. So, and you guys just shout if you need me to speak up a little bit. I'll try to have some volume here. I'm in the office, so somebody might throw a shoe at me and tell me to be quiet, but I'll do my best here. So I wanted to zoom out a little bit today and not really focus intently on any one specific case or system or taxon or anything like that, but it struck me that maybe a lot of folks don't know exactly what DEQ's role in the HAB task force is. I'm going to try to fill in those details a little bit. This thing that you're looking at is crazy because I came up with it, and maybe it's a view into the dark reaches of my brain, but yeah, myself and the team of water quality folks came up with this, and then we kind of got consensus from across the state. It's our sort of decision-making flow chart for how we investigate a bloom. When you're on site for the first time, how do you go about determining whether it's a HAB that should be sampled or, you know, a potential harmful algal bloom that should be sampled with lab investigation? What do you do? What observations do you have? The details are not super important other than I will provide at the end of this a bunch of links, and the ones that are DEQ documents like this, if you have feedback, please send it along with this. These are always working documents that are living and need improvement constantly. So basically, DEQ serves as a co-lead with Virginia Department of Health on the HAB task force. There's now an MOU, as Margaret mentioned, for that, a memorandum of understanding of exactly what that means. Functionally, what we do largely is the field stuff. We conduct the majority of HAB investigations, almost entirely, all the HAB investigations in freshwater that are reported through the VDH online HAB reporting portal. So there's a report of a potential bloom from the public generally. We go out and we do the HAB investigation, or observe conditions that don't indicate a bloom and maybe don't do a full-fledged investigation. And how that occurs is based on this big old flow chart here. Our practices, you know, we participate, we sometimes lead those core meetings that Margaret was talking about, VIMS and ODU and VDH and us, other groups as invited. Those are like the day-to-day activities of the HAB task force, specific cases, specific protocols. What do we do about the sample bottles? A lot of it's kind of boring stuff to outsiders, but it's cool to us, and it, you know, keeps things functioning. So we're an active member in those, and we take turns kind of coordinating and leading those as well. We develop DEQ protocols. So this protocol is similar to, but not identical to, several others. I believe ODU has one. but this is a DEQ protocol. So our internal stuff, as well as our internal HAB data evaluation, the functions that are DEQ, we are the developers of those protocols. We solicit feedback and input from HAB task force partners, but the HAB task force is sort of a consortium, but state agencies like DEQ and VDH have internal protocols, and those agencies are sort of the ultimate drivers of those. So the task force doesn't develop DEQ or VDH protocols, but they can inform, give technical guidance on that, and then we in the agency develop our protocols based on that guidance. This flow chart is one of our big things. I'll give you a link to this, as I said. It's got that flow chart. It has a whole bunch of notes here on different steps, who to contact, what to do. It's very long, but very detailed, covers those initial investigations that needs to be changed and adapted for ongoing cases. We have field data sheets here. The regional field staff modify these depending on the case, but it's just, a way to lay out everything that we would like to have, all sorts of parameters here. We don't always have, for instance, blue-green algal probes or chlorophyll probes, but if you do, or turbidity, but if you do have those, here's how you record them. So that's sort of this thing. Other functions that we do along those lines. So Virginia Department of Health does the health advisories, as you heard, but we at DEQ are responsible for the assessment. So for the first time in 2022, water bodies could be listed for non-attainment of the recreational use standard. And those were based on the issuance and persistence for a period of time of the VDH health advisories. So they weren't based on a threshold number, but VDH uses threshold numbers. They then say yes advisory, no advisory, and our assessment results were based on that. So this is our integrated report page. It shows the 2022 report here. You can get the guidance about how that was developed. Pretty soon, in the next month, we'll issue guidance for the 2024 report. That'll go out for public comment. So again, feedback through that mechanism, or your methods for assessing here are good, bad, or indifferent on them, whatever. That public comment period is about to commence. So those are kind of, as I said, VDH does the advisories. We then do the assessments, which are more like a long-term evaluation of not is it healthy to swim in the water today, but is the water body attaining its recreational use, designated use, or is it a water body where it doesn't support that use, and therefore we would list it as impaired. And HABs are one of the things, VDH's advisories, that we can do that based on. We provide technical feedback and then policy input, but not direct decision making on other task force items. So the VDH recreational HAB thresholds, we provided input on those, those thresholds and their articulation and the guidance. Is it clear that but the final decisions there were VDH. Practice on how to quantify HAB toxins and potential HAB taxa. Again, those are laboratory things. ODU does the vast majority of the laboratory functioning for the HAB task force. We may provide technical input on those things and suggestions, but then ODU ultimately makes the decisions about how samples are evaluated for taxonomy and toxins. And then I'll say other duties as assigned, and that's mostly by the governor and the General Assembly. You'll hear a little bit today. I'm not going to go into great detail, but there have been a number of General Assembly bills, House bills, that have driven what we do at DEQ and consequently VDH and universities regarding harmful algal blooms. They're more and more on the radar. There are General Assembly members concerned, and then there are constituents that have influence on those General Assembly members, and there's kind of growing concern for these. Probably a good thing. It probably means there's going to be more and more support for work to do with HABs and understanding the problem a little bit more. So the first one of those was House Bill 1800, I believe, and there's a lot of words here, but I'm showing you that now. Stuff struck and stuff included. The very last item here that I'm showing, and I don't even know if you can read it clearly, there's a lot of text here, but we were charged as the lead agency in collaboration with the Virginia Department of Health and the Department of Agriculture to achieve these objectives here. Basically, it says the location, frequency, and severity of harmful algal blooms in Virginia waters, the factors that lead to the formation and occurrence of the blooms, and then plans and strategies for state agencies to lead or support appropriate mitigation efforts. Now, here's the entertaining part. This is a budget bill. If you go up here to the budget and you look for the money allocated to DEQ and VDH and partner agencies for that work, you will see no line item. This is a budget bill with no actual budget allocated, which means we just do the work. The other thing is this was issued in its final version, I believe in March of 2021, and then there's this date of September 1st, 2021 to submit that report. So we have $0 and we have much less than a year to complete these rather lofty 3 objectives. So what we did, I think, is a great job, and it does say, to be fair, it says findings and recommendations related to. So we put together this great report. I was very surprised at how much information we gathered, especially Todd and Margaret at VDH pulled together a great deal of information on advisories, their occurrence, where they occur, over what years, and I'll give you the link to this report too. And I'm having trouble scrolling, I'm way down here at the end, but it's a big old report. I think it reads pretty well. Harmful algae blooms in Virginia, Commonwealth of Virginia report, September 2021. And we were careful to say, look, we're, as a scientific investigation of the location, frequency, magnitude, duration of blooms in Virginia, this is biased because it's our sample set is targeted on reports from the public. So we don't have a sort of probabilistic data set to say this is how they occur and how severe they are. But we do have a lot of data. And I was, again, particularly impressed VDH's component. So maps on marine biotoxin occurrences, potentially toxic marine algae. There's a whole series of maps like this that sort of summarize over this period, sort of 2017 to 2021, where we had data at the time how they occur. So this is potential HABs reported, so the number of reports, and you see we're highest in August, July and August, which you might expect the latter part of the summer period. July to September is the hot period. Reports by county, Louisa and Spotsylvania top the list. Spoiler alert, that's Lake Anna country, so that's where those reports come from. But how they lay out by county. There's also advisory issuance maps. Let's see, so there's potentially cyanobacteria, different components here. So very thorough report. We also detailed in a literature review of the potential solutions, mitigation and treatment strategies. There's just a great deal of information in here, and I thought it was a pretty good synopsis at the time to address those variables that may be causative factors. We produce a trends report. So the last one detailed in this report was water quality trends from the period 1997 through 2016. So long-term 20-year trends, degrading or improving or no trend with respect to a bunch of parameters such as nitrogen. So that's laid out here, and the degree to which that may or may not relate to HAB events isn't analyzed in here. We didn't have a data set appropriate, again, that was unbiased for analysis of X versus Y, but we did summarize that information, so it's laid out for the reader here. So I think this is a great resource if you hadn't seen it, and I don't mean to highlight all the details in these, but just to show you that it exists, and I'll certainly provide links to all of this stuff. The next House bill, which is this year, is it's up here, blah, blah, blah. There's a whole bunch of stuff. We don't even need to read this, but I'll provide you the link to it. But it essentially, go back to the flow chart because it's at least a little more interesting. House Bill 30, issued in 2022, allocated $3.5 million. The wording was very simple in this case, $3.5 million for the study of harmful algal blooms in Lake Anna and the Shenandoah River. It didn't indicate how to study, what the real objectives were. We have kind of refined those objectives in a way that sort of is functional. Our objectives really are to quantify the relationships between potentially harmful algal blooms and the environmental factors that cause them, and then to provide evidence on potential solutions, potential mitigation factors. So we're interested in a little more, not just study, but what does that mean? We're moving quickly out of the proposal phase and into the execution phase for those things. We've engaged as primary partners, the U.S. Geological Survey and the CPRB. Well, I'm not gonna get it. The Intercoastal Commission for the Potomac River Basin. Mike Seligman can correct me if I messed that up, but as primary partners in that project. And I think you'll hear a little bit later about the technologies and sort of strategies for monitoring. To again, sort of primary goal is correlate those environmental factors to the HABs, when, where, and how and why do they occur in those two systems, which are distinctly different. The Shenandoah, we're focused on benthic mats that may potentially produce HABs, and in Lake Anna, it's largely the planktonic blooms. So that's a 10,000 foot synopsis of where we are and what we're planning for 2023. I have a feeling, I don't know, it's just gut feeling that maybe we're in for a tough year. If the summer is warm and wet like the spring was, we could have a problematic HAB year. In the impoundments, we see these blooms in warm, dry periods after a rain. So you get those surface inputs, and then it gets sunny. The water level drops, and the water is warm, and it has inputs of the materials, the nutrients and sediment that support those blooms, and they take off. So in addition to these studies, I'm sure we will be busy responding to cases that pop up. I think that's really it for me. I was pretty quick, I know, but I'm glad to take any questions.
Anybody here have questions for Drew? Or online? Question. Are we gonna have, look at the agenda, unfortunately. So we're gonna have some follow-up with ICPRB talking about following up on, Drew said the project that they're sponsoring. That's PRB.
Yeah.
You're gonna stop here.
Yeah.
Okay.
Yep.
All right, thanks. Are there any more questions? You're really rocking up. Thank you. Well, it's obviously the next. At least something, the money was expensive. Always do that. Good.
I haven't bought undergrads in a while, but I like the distribution in the class right now. Everybody's clustered back by you're ready for the builder. If you never grow out of something.
Never.
Well, while we're looking at that, I'll go ahead and introduce my speaker is myself with the ICPRB. Mike has worked with ICPRB for 10 years, is the executive director for aquatic habitats. He's worked on a range of Potomac Basin algae investigations, such as nutrient thresholds relative to primary production, water quality trends as predictor of problematic algal blooms, the assessment methodologies of algal blooms. What is your PowerPoint?
There we go.
All right.
Thank you.
Now, hopefully everybody online is seeing the PowerPoint properly.
Well, So, yeah, I guess, it's nice to be here in front of people again. So I'm gonna be talking specifically about, another basic freshwater HABs, flowing systems. You lake guys, clock out for a little bit. That's not gonna be this talk. So the first thing, and thank Drew for propping me up on this. Who is ITPRB? We're the Interstate Commission on the Potomac River Basin, and, our mission is to protect and enhance the waters related to the Potomac River Basin through science, regional cooperation, and education. This is the big thing here. We've been around since the 40s. We just had our 80th anniversary. And the main things I want to highlight is that we're non-regulatory. We are basically, strictly a research-based institution. So we have a couple niches in this. We're reaching out. A lot of times, state requests are prompting the research that we're doing. We're kind of, in a lot of cases, the tip of the spear. If there's something that there aren't methods developed, there's maybe a new water quality frame or something that a novel way of looking at a data set, accumulating data sets. That's kind of where we've made our niche. So we're here to help the Potomac Basin states. And again, I hit back to the non-regulatory part because I'm going to do my absolute best. There are certain words that we just don't touch. Assessments and thresholds, things like that. We provide the data and let the discussion happen outside of our agency. So again, some of the things that I just kind of wanted to highlight: the methodology development, mortuary analyses, the research coordination and outreach. That's the last thing I kind of want to highlight. A lot of times we're a small agency. Right now our office has around 20 people in it. But we're very good at, positioning ourselves with identifying partners in projects, universities, federal and state agencies, and we try and make projects that sort of highlight everybody's strengths. So there's the sales pitch for ITPRB. Now the timeline of how we've been involved in HABs, I argue this a lot. What are we calling a HAB? I start every meeting with that because there's this nuisance green algae issue that has emerged from states like West Virginia. There's toxic human contact, human health issues. A lot of times what makes the press are the toxic issues, and the nuisance things are kind of more of your general river users complaining about their fish hooks getting fouled and they can't take their canoe down the river and things like that. So because we don't have a lab that can process a lot of toxins, we don't have a lab, we can do some taxonomic ID, but generally we're trying to find labs to coordinate with. We're very narrow in what we can do to help here. So we started in 2012 with looking at the South Branch, Potomac and the Cajun Rivers for West Virginia. I'm going to go into that as one of the kind of highlighted examples of sort of interesting nutrient chemistry and how that affects the big picture. 2014, 2015, we worked with Virginia on their longitudinal survey methodology. That's still yet to be solved. It's something that I hope we can work on in the future. The whole point, grab sample versus understanding a living, breathing thing over a large area. I always have this concept of grab sample. 2016, we had the taxonomy workshop. That was just a real quick rundown of highlighting who are the worst players in the basin and what to look for when your field crews are just kind of passing through. So this wasn't into the weeds. This is just a real quick high level pass through. And then I put predicting blooms, but really I like to refer to it more as looking at nutrient, non-nutrient factors and the risk of blooms, because predicting blooms is incredibly difficult. There's way too many things. Predicting is one of those taboo words, and I thought I'd jump back and change that. But here we are. 2020, we worked with EPA on their nuisance algae task force. We brought some new technology into that, like the Pectenotorch, met with different state agencies and talked about what their field methods are capable of producing, and what are their strengths, what are their weaknesses. And then we've been kind of on call for the last couple of years, helping Virginia with their HAB response. That's been some general support with literature review. We've taken some samples with the Abraxas work that was mentioned earlier. Taken some of the longitudinal through some boats in and did some scouting type work. But really up until 2021, toxin analysis wasn't anything that we were involved in. Now we're kind of directing where that work can go. So the first one is the Tale of Two Rivers. There's a couple names that are familiar on the Zoom call. Sorry guys, you've probably seen this before. But the South Branch and the Capeman are a really good example of relative proximity within the basin, but very different systems, and why we need to consider this oversimplification of nutrients called algae. Yes, but also as we're scientists, it's more complicated than that. So when we went and surveyed these two rivers to West Virginia, we took these physical qualitative assessments. We did a visual algae transect. You know, one of the issues with the transect is you're going to the worst part of the bloom and reporting the worst single point across the river. Really, it's not that descriptive, 60 miles of river. We took chemistry at each of these points, your nitrogen, different forms of phosphorus. The ones that are really important to point out are your total alkalinity, calcium magnesium index, as well as everything that kind of can come with the YSI. And you know, if on the I have axis over there, you have total phosphorus on the bottom, or this is Capeman sites and South Branch sites, the FB here. And if you were to look at this, where's the algae bloom? These are locations. It's a pretty easy answer, right? Like a lot of nutrients, you know. So the elevated phosphorus was from a point source. That was the Moorfield wastewater treatment plant, 2012, 2013. These were predictable algae blooms, very abrupt start to the bloom with a tailing effect. It was predictable every year. And once the wastewater treatment plant was upgraded, we've lost the chemical signal. So it's that, you know, pre and post, this is a success story. And as far as I know to this day, the blooms really haven't come back. There's occasionally little cyanobacterial tufts that show up, but you go anywhere in Potomac and you'll find this kind of low density tufts. So a success story. That was the same site a year later, before and after. And again, so these are, I know this is a HAB task force. These were mostly filamentous green with cyanobacteria co-occurring in these areas. So it's predominantly filamentous green, but as you move through a season, there was a succession of different species of green algae as well as cyanids. The Capeman River is very interesting, and to try and wrap this up to a single slide, we don't really get a nutrient signal. It's always just above threshold of detection, and depending on the flow year, depending on the timing of spring flows, you have a invasive hydrilla species that will dominate, fill the water column. And that's generally linked to a higher flow year because the vascular tissue allows it to establish in the system. If it's a lower flow earlier season, you start seeing the filamentous green like hydrodictins set up early in the year, and then that kind of turns into the problem child, the Phytoplankton. And that'll fill the column. And in some years, when the water's gotten very low and temperatures have rose, we started getting cyanid blooms sitting on top of these filamentous green patterns. So if we were looking at nutrients alone for this 3 to 5 mile reach that is just absolutely choked with production, you know, the answer's not so clear. So we took all of our kind of qualitative, you know, a 1 to 5 scale of total production. So that was 1 to 5 for blue-green algae, 1 to 5 filamentous green, emergent, submerged aquatic vegetation. Added them all up and looked at, compared them against, I guess, the environment, the water quality that was there. And what you see is this kind of classic wedge. As production goes up, you have less of a nutrient signal because these things are taking in the nutrients and the water chemistry is reflecting the biology of the system. In areas that might be higher flow, or there's some other limiting variable that's putting down total production, you start seeing nutrients being more elevated in certain areas. So this is referred to as our classic wedge. And this is a point source influence system. So again, this is the South Branch, the successory, right? The one thing I wanted to highlight, James Summers, a chemist in West Virginia, pointed out this kind of Goldilocks zone between alkalinity and hardness. And without getting too far into the chemistry, it's really fascinating. Basically, it changes the binding affinity of phosphorus. And with that, you actually create this positive feedback loop where the natural respiration photosynthesis cycle that takes place, we see the pH go up and down, and it seems like there's, as the system gets further and further out of balance, it's crossing a threshold that's making phosphorus more bioavailable. It's feeding the organisms, they're growing more, the whole system kind of goes out of control, and so we have a temperature issue come autumn. So for the South Branch, basically 99.5% of these sites were outside of this alkalinity criteria, this window, the Goldilocks zone. But when we look at the Haven, we have Lost Star Wedge, and basically we're at this kind of like vertical profile of our phosphorus value. And again, there was a lot more chemistry. This is me just trying to run through it, hit the big player. 78% of the locations that we sampled in the Haven fell into this Goldilocks zone, and they were able to kind of correlate with this total production. So it starts looking like there are nutrients in the system. We know that because something has to be feeding this level of biomass, but we're not able to detect it well. So we kind of took a step back and we said, well, is there, we'll call it risk. Again, I don't want to call it anything predictive, but are there areas across the Tennessee Basin that might be higher risk for blooms, higher production? And if so, what are we connecting to the watershed? Is it surface runoff in these environments? Is it groundwater transport, things like that we need to consider the environment that we're in? So this was the first time period we looked at, a 10-year window and a couple of years following. And because calcium, magnesium, the alkalinity parameters aren't samples continuously like across the Chesapeake Basin, the Potomac Basin, it's kind of our best grab of pulling storage data, trying our best to clean it up and see if there is a story here. And what's interesting is that the Haven kind of pops out as one of our risk areas. I guess the Shenandoah, South Branch, there are certain areas, and this is just the Hug 8 level, that seem like they may be higher risk based on base geology. So something that we kind of started diving into, like, all right, we know that there's this predictable 5-mile reach in the Haven. Let's look at every potential nutrient source that we can link to surface flow and see if that's going to this 5-mile flow. We should be able to find a creek, look at the LiDAR data and say, this is where our flow lines are. We should be able to look at the MLCD data set and knock on doors, look at farms and say, identify where potentially these nutrients are coming from. If this is in fact a point source happening within 5 miles year after year. And what we found was that this LiDAR work and the land use, we actually went out and with help with West Virginia, took water quality samples throughout the year at all these creeks that should have been the points that would be delivering the nutrients. We never had any sort of detection. So taking the next step forward, USGS did some great work, Dan Doctor, took a LiDAR data set and looked at karst depressions, areas that are indicative of limestone geology, that sort of groundwater transport. This area in the Haven Valley isn't notoriously known as the karst system. It's actually kind of a pseudo karst, and I'm not a geologist, so I'll get angry emails later. But what's interesting is that we put out, so in the next 2 years, we're going to be putting dye into this system and trying to see where the groundwater transport paths are. In the last year, we did background monitoring just to see if there was anything that was going to throw a false signal to mess up this upcoming project. And we accidentally came across one of our wavelengths rising right around the bloom site. And we're trying to figure out where that's coming from, but we sampled upstream of the bloom, in the bloom, and all the streams around it. And it looks like there's water upwelling in this system. So it's just a pathway that I don't think we're exploring enough for this HAB discussion. Now, it's something completely different, longitudinal methodologies. Right now, there's a lot of good point surveys. West Virginia has their method, Virginia has their method. I almost threw up the table that we had put together of like 40 other states and their method and what they're good at. Everybody has something that their field crews are doing, whether it's a narrative, yes, there's algae, there are cyanos present on the field sheet, but the quality of that data and what they're reporting, very inconsistent. And to try and go back, a lot of times it requires going back to the data sheets because the digital data sets are leaving out those handwritten notes. So it's very difficult to actually find historic algae data. So the point survey methods exist, but again, they're snapshots. They're missing that pH, the DO, this living, breathing system. We go into what continuous surveys, the longitudinal methodology, and we did this years ago with Virginia, where we went through and did transects at set points going down a river. We did randomization at set distances. We did a totally randomized approach. And basically what it comes down to is you do a power analysis of what you can describe, how many samples you need to truly describe what a bloom, the little details, the different types of algae that are present. It's a huge effort. So there's always some level of compromise as to what we can really describe over a long reach, the 52 miles to the Shenandoah. Go out there, put boats in. We're limited to 10, 50 miles a day in a canoe with narrative descriptions of what's going on out there. You start adding algae sampling and toxin sampling, water quality, all these things. The distance shortens, and we start getting back to this point survey. So one of the things we looked at, let's speed up the process and use digital photography. And one of the main things we found out pretty quick, the diminishing plane is the real thing. You can take a picture at ground level and kind of capture a bloom. We can pull out the types of algae, where the growth is, but ultimately, we don't know what's on the far bank. It ends up being just labor intensive as anything else. You do smaller, like server sampler size quadrats and pull out coverage, get an idea. Again, we're kind of limited. So that's the issues with, we don't really have a good way of characterizing water issues right now. On to the next, I have 2 minutes left, I'm going to fly through this. There were toxins reported in the 52 miles in 2001 in the Shenandoah. We were prepared to kind of act and help describe what these blooms look like, and then Hurricane Ida hit. And instead of claiming that the hurricane hit, we're like, well, there's still the potential for a lot of toxins there. This is going to be a scour event. Let's go out and we alerted another part of our agency, we alerted our water utility relationships that like, maybe take samples, keep your eyes peeled. We took some samples, sent this off, and found that we really didn't get any toxins in the water column during the scour event. But what was interesting was we were able to use our spill model to predict the travel time of when these toxins would be in certain places and alert the system as we went downstream. But as any good science should do, intercellular toxin concentrations, where did they go? Are they just diluted away? Mechanical lysing, the dilution factor stuff. A lot of questions came out of this. So that end of the year senescence and flush, we don't really know a lot about. And then finally, we're helping Virginia with this, their HAB response plan. This is our, a map of kind of where all of the filamentous and HAB reports have happened. Talk to Virginia about this stuff, over the story better than me, right now. So finally, the needs of the basin. We need to really start considering non-nutrient chemistry, and explain these blooms, which means that the regular ambient monitoring strategies probably aren't enough. Looking at things like iron and alkalinity, the underlying geology, we should be working towards a rapid large scale assessment tool. Satellites would be nice. I feel like it's been promised for higher resolution for years and years. Someday they'll be here, not this year. Drones, I think, are really for flowing systems and the scale of where we're operating. I think that's the future because we can get archival ortho images of what's happening in the river. We can overlay hyperspectral imaging, which there's a lot of work that's being done on the West Coast that they're creating these hyperspectral banks that start getting the actual IDs. I think a very good fair target for step one is let's just see if we can differentiate between filamentous green and cyanos, and then we can start working in from there. Photogrammetry, the triangle of two images, we can start looking at substrate, we can start looking at flow velocity from these mosaics with drone imaging. And finally, add some context. I think we need to do passive sampling to understand toxins in the water column versus this intercellular vat sampling versus grabs, because we don't know the timing of when these toxins are being released, and we don't necessarily understand the cycle of how toxins are being processed within the cell. And with that, any questions? I think I went over, I'm sorry. That's it.
We started you late, so if there's questions, please ask.
So that last slide that had the was talking about what you're doing now.
It's what we're trying to bring to the basin. So there are projects that want to go back one.
Well,
Is that here?
before that,
Okay.
yeah, that's the one that's building off of the.
No, this was actually, that was something that was submitted, beginning of last, or I guess it was finally in July, but we worked on that spring of last year. And that was just a lit review kind of, looking at what other states do, their mechanism for decision making. A lot of what Drew was talking about.
Yeah.
I haven't talked anything about, the Shenandoah that's not coming here, other than those technologies would be nice to get there. Oh, that's great.
We don't have time.
Let's talk to you.
We'll talk to you.
Yeah.
Okay, yeah.
Anybody else? There any online questions?
Thank you.
Thank you. Up next, we have all four of the labs are gonna have a presenter. We're gonna start with Reece's lab, and the speaker is Sylvain Gaillard. He's a postdoctoral research associate with the Michigan Institute of Marine Science, Aquatic Health Sciences Department. Sylvain work at VIMS mainly focuses on fish physiology of pharmacological species such as Biotoxin, and Proledinium beneficum, as well as other aquatic organisms, i.e. eastern oyster. Thank you.
Hello everyone, I'm Margaret at VIMS. And today I'd like to share with you results of an exposure experiment we've done on the Crassostrea virginica exposed to Alexandrium modulator. And we specifically focused on toxicity pathway, pathology, and gene expression. I will start with a brief introduction on Alexandrium modulator, the type of flagellates, the bioluminescence, checkpoint flagellate with a tropical and subtropical distribution that's been found in South America, Central America, North America, and relatively warm water. It can form dense and large bloom with discoloration of water, as we can see on this picture, especially in the south of the Chesapeake Bay. And in the York River, it has been shown to bloom almost every year since 2007. So this is the chain of Alexandria modulator. And this, the flagellate can produce toxigenic compound, the goniodomides, which negative effect on aquatic organisms. And here is an example of toxin, the goniodomide A. On this map, you can see the occurrence of negative effect on aquatic organisms caused by Alexandria modulatum group in the US. And for instance, research on fish and shellfish mortalities exposed to bloom of Alexandria modulatum in the Gulf of Mexico and Florida. And also the Chesapeake Bay epithelial erosion has been observed in Crassostrea virginica exposed to bloom of Alexandria modulatum. And in a study, researchers found accumulation of goniodomide A in bivalves exposed to bloom water of Alexandria modulatum. Laboratory exposure studies corroborated the observation that has been made in the field. For instance, fish mortality in larvae, juvenile, and adult fish have been observed when these organisms were exposed to senescent culture of Alexandria modulatum or to lice cells, Alexandria modulatum, as well as negative effect in shellfish. For instance, shell closure, decreasing currents rate and validate, and as well as mortality has been observed in bivalves exposed to senescent phase, cultured senescent phase of Alexandria modulatum, as well as mortality in oyster larvae and clams larvae when they were exposed to lice cells. So it's interesting to see that in the literature, when researchers found negative effect on bivalves and fish, it was almost only where the animals were exposed to cultured senescent phase or to lice cells. You can think that there is an effect of extracellular compounds, which could be the goniodomides. In our study, we wanted to know what effect does a low concentrated exponentially growing culture of Alexandria modulatum in clean culture media have on the physiology and gene expression of Crassostrea virginica, and what are the kinetics of accumulation, compartmentalization, and degradation of goniodomides. So we performed a 96-hour exposure study with the monoclonal culture of Alexandria modulatum, which has been isolated in the York River in 2007. And this strain produced a high amount of intracellular goniodomide A, more than 1,000 picogram per cell of goniodomide A. It also produced goniodomide B, goniodomide C, and goniodomide A sequencing, which are isomers of goniodomide A. And it's interesting to see that this strain produced around 25 microgram of goniodomide A sequencing in extracellular compartment. So in this experiment, we used exponentially growing culture of Alexandria modulatum responded in clean culture medium before the experiments. So we wanted to ensure that the extracellular compartment, the culture medium, was almost free of bioactive compounds. We also used, we choose to work with Crassostrea virginica oysters provided by VIMS naturally, and these oysters were acclimated and started for 5 days before the experiment and acclimated to the lab condition. So the experiment consisted of a 96-hour exposure experiment followed by 96-hour deprivation of the oyster with a daily renewal of seawater and air. And so we exposed the oyster to 3 different treatments. The first treatment were oyster fed with Alexandria modulatum, around 600 cells per milliliter. So every day we change the water in the tank for the oyster water, and we add new edge. The second treatment were oyster fed with non-toxic phytoplankton, and the last treatment were oyster unfed. So we only change the water every day without addition of phytoplankton. So after 96 hours of exposure, we allowed the oysters to degrade to the diet of non-toxic phytoplankton for another 96 hours. So during this experiment, we sampled water to do some phytoplankton cell enumeration and purine rate population in the oysters. We also sampled feces and pseudofeces of oyster for observation under the microscope. We assessed the mortality of oyster by response to physical stimulus, and we sampled oyster different times during the exposure and the deprivation phase. So we sampled the oyster for weight and measurements, and we also checked the oyster and do some dissections of gills, digestive glands, and remaining tissues For toxin quantification, in collaboration with Mondial Laboratory in France, for gene expression analysis and paraffin estimation. So the main result that we observed in this study was that was no mortality in none of the pretreatments. In addition, we observed a positive clearance rate in oysters exposed to Alexandrium moniliatum, which suggests that the oysters were able to filter the Alexandrium moniliatum. And when we look at the pieces and pseudopieces of oysters, we observe live Alexandrium moniliatum cells in the. We did not observe any tissue pathology. So on the left, the image of the left is gills, and on the right is digestive glands tissues of oyster virginica exposed to Alexandrium moniliatum for 96 hours. And you can see that both of these tissue exhibits some healthy histology. Let's now have a look at the toxin quantification. So on these plots, you can see on the y-axis the toxin concentration according to different time of exposure, so 6 for 96 hours, and different time of diversion, 96 hours. This plot here is for oyster gills. Here digestive glands, here remaining tissue, and the last one, all individual, means that we have normalized the toxin concentration of the tissue according to the weight of each tissue and the weight of each oyster. And the different colors represent the different toxins. Vitamin A, A sequencing, B, C. What we can see first is that oysters were able to uptake the rhododamines in less than 6 hours. So in each tissue, you can see that you can observe toxins. While we observe some viability between the oysters, so you can see it's relatively big around. The maximum concentration of gonadamine A was observed after 6 hours, and it was around 2,500 micrograms per kilogram of shellfish gills. So we observed a lot of toxins in the oyster gills. And at the same time, we observed 21 and 4 times less gonadamine A in digestive glands of the oyster and remaining tissue, and 3 times less when normalized by weight in all activity. After 24 hours exposure, the trend of decrease in concentration in gonadamines in gills and remaining tissues, while you observe the trend of accumulation of gonadamine A and sequence in digestive glands to reach around 429 micrograms per kilogram of gonadamine A sequence in digestive glands. And interestingly, we can also observe the biotransformation of gonadamine A to gonadamine A sequencing in digestive glands. So you can see, for instance, the gonadamine A or in gray, and gonadamine A sequencing in blue. So there is a transformation of gonadamine A to gonadamine A sequencing. So the depuration, we can see on this graph that the oyster were able to quickly depurate gonadamine B and gonadamine C because it was not detected after 24-hour depuration. However, we still observe gonadamine A in all the compartments after 96 hours depuration. So for instance, in all individuals, we normalize by weight, we observe around 4 micrograms per kilogram of tissue of gonadamine A per tissue of oysters, and also observe the gonadamine A sequencing in the digestive gland of 6 hours. So this plus represent the gene expression of oysters exposed with Alexandrium moniliatum. So if y-axis is the relative quantity of gene expression according to different genes and different compartments, so genes or digestive glands, and the different treatments. In pink it's Alexandrium moniliatum, and in green it's unfed. So what we can see here is an overexpression of genes involved in redox cycling in digestive glands. So the genes are glutathione peroxidase and glutathione reductase, suggesting that oysters were subjected to oxidative stress. We can also observe an overexpression of genes involved in actin synthesis, and it's very interesting because the gonadamines have been shown in the literature to target actin filament in some tissues, and it can enhance synthesis of actin or enhance this actin. So this is what we get observed here. And also an overexpression of genes involved in apoptotic process in animals. After 96 hours of depuration, so the second graph, and we can see a third expression of redox genes, suggesting a low antioxidant capacity of oyster at that time, which could be vulnerable to Alexandria mediator or other stressors. We also observe a low anti-inflammatory low expression of anti-inflammatory genes such as GAT1 or stress genes such as alpha, suggesting that oyster may not be stressed at that time and were maybe able to do some detoxification. So I will finish by a short conclusion of this study. So first of all, we show the toxin profile of the local strain of Alexandria modulatum, which was a high producer of guanidamine A with more than 1 nanogram of guanidamine A per cell. We did not observe mortality and visible physiological stresses in oysters when they were exposed to low concentration of Alexandria modulatum, which could suggest a lower toxicity of Alexandria modulatum cells compared to extracellular metabolites. The oysters in this experiment were able to uptake, compartmentalize, accumulate, biotransform, and degrade several guanidamines. And they were also able to uptake more than 2.5 nanogram per kilogram of the toxins in their leaves. And after 4 days of depression, we have still observed some guanidamine tissues. We observe effects of expression on genes above the important physical process, such as redox cycling, as I mentioned, and which means that oysters were subjected to oxidative stress. And it's interesting to see that in further experiments in the exposure of oysters to Alexandria tamaracid, which is a producer of toxins, they observe the same effects, negative effects. And here's some implication of this study. So it could suggest that oyster, so we show some physiological effect on genetically important species of the oyster, and it can be an important stress during dense and large bloom vegetative pain, especially because the food can last for days, weeks, and here we only did a 4 days exposure, but it could suggest that if we expose oyster on the field for several days, they could accumulate a lot of toxin and they could be stressed by this. And for some perspective, it would be very interesting to study the long-term effect of chronic exposure to extracellular and toxins, as well as to study the transfer of toxicity. So we show that oysters can accumulate toxin. It can be interesting to know if these toxins can be transferred to natural predator of oysters, which can be the crab here in the bay, as well as to study the effect on sensitive vanilla stages of oysters. Thank you.
We have, and you went a little, we have time for questions if anybody has one.
There's no online question, but I think it's part of the next steps. But Paul asked, what is the long-term effect on growth of the oyster? Is it permanent or is it short-term effect?
On growth of the oyster, I didn't study here the effect on growth of the oyster, but I remember, yeah.
Seems to be short term from what we've seen. They seem to recover their growth rate after the exposure. If you look at the growth rate graphs in the field, recovery pretty much after a few weeks of the slower growth rate.
Thank you. Juliet's.
Yeah. Yeah.
You know the magic. I'll go ahead and do the introduction while we're getting it switched over. Up next, we have Juliet Smith, who's the associate professor with the Department of Aquatic Health Sciences at the Virginia Institute of Marine Science. And then Juliet's going to be presenting for the Smith Lab at SIMS. They're investigating over chemistry, ecology, ecotoxicology of bioactive compounds synthesized by harmful algal blooms in freshwater, estuarine, and marine environments. It's up there.
It looks like it's the right view too, yes?
Yeah.
Okay, let me see if it works. Hold on. Excellent, perfect. Okay. Thank you so much, everyone. Happy Friday. And thanks for the invitation to just tell you about a few things that my lab is working on, and we're going to continue into 2023. Do you want me to pause, Kim?
No.
Okay. Oh, no, I got it.
Like this file, it's split, it's, oh, it's phenomenal, I'm sorry.
I think that the box is too big.
There.
Oh, yes. Oh, you're trying to like, oh, no, it's older there, and then it's not. Green. Yeah, but if I let go.
Can you minimize the size of the window?
Oh,
Yeah.
I'm tiptoeing back, okay.
Okay, I'm good. All right, so the three things I'm going to be telling you about, I'm going to quickly walk you through what we're working on into 2023 with the IFCB and our toxin monitoring, which right now is focused off the VIMS pier at the mouth of York River, and then new passive sampler that we're hoping to really start with in 2023 with improved microcystin and domoic acid affinity, and then some toxicity of phytoplankton biochemicals. All right, yep, looks like you can see it. There it is. All right, so moving on to the IFCB HAB monitoring. So we've been doing this since 2017 through the present. Again, that's on the VIMS pier. That IFCB data is now about to be displayed on the HAB hub, which is at HUI. And if you ever want to check that out, that would just be, you can Google HUI and HAB hub and it will come up. That effort is led largely by Mike Brosnahan and some collaborators he has on another NOAA project. And then we get to kind of piggyback on that, which I'm really excited about. Because up until now, we've had some security issues that have restricted us from being able to display real-time data from the IFCB via VIMS website. So this is our way to finally get all that data right up on the web for everyone to look at and use and download. Please use. That we're hoping will be up by May 1st and available to everyone. Right now, we're just going through the last phases of that. Currently, we are using a convolutional neural network, or what we call CNN, as our classifier. And that's focused, we have CNNs focused seasonally, and some CNNs that are focused maybe to focus on different species of interest, different HABs. And by working with the CNNs, we can kind of come up with more very focused models, as opposed to before where we tried to have like one random forest that did everything, and that wasn't really working as well as we wanted. So we've moved over to CNNs. I wanted to make sure I said this out loud in this forum. Please, we are very happy to share our training images. Vanessa, Wade, Marta, previous undergrads and graduates have worked really hard to build these image classes that range anywhere from a few hundred up to thousands of images. So please, if you'd like them, send me an email and we'll figure out how to get you those training images to either work with or add to your existing classifiers if you're interested. And then the way that we're using that is Vanessa's working towards some ecological modeling to try to do some abiotic and biotic predictions of HAB species of interest. And Wade's going to be moving probably more towards ecological networking, kind of who's there when the organism of interest is as well. So who's there before, who's there after at the same time. So that's coming. We're working on that right now. This is a picture of the HAB hub. I just wanted to show it to you really quick. So as you can see, it's a HUI Gulf of Maine Nauset Marsh product originally. But down here at the bottom of the map, we're going to be adding in the VIMS pier. But you can see that essentially you could go in at any time and see live data from any of the IFCBs that are part of the HAB hub, and you essentially just select what species of interest is yours. So it's a really cool tool. All right, so moving on to new passive sampler. Okay, So please do not take this as I am not supportive of SPATs, because I am a big fan and user of the SPAT. However, there are some caveats that are becoming more obvious as of recent. So new evidence suggests that the more polar toxins, so that's going to really be your domoic acid. Some of your microcystins fall into the more polar category. Those are being found to desorb usually within a day, sometimes hours, from a SPAT in the field. And that was recently shown by Reece and his group over at the US HAB Symposium in October of 2022. That doesn't say that a SPAT is useless for those toxins. It just means that it's most likely more of a snapshot. So what was probably there within the last 24 hours versus what we like to use SPATs for, which is kind of this accumulated time integrated measurement of toxins over time. But then with the detection of microcystins and domoic acid in oyster tissue, which Sarah Peace showed back in her dissertation in 2021, we really wanted to kind of focus in more on this. Well, now that we know that microcystins and domoic acid are something we should be keeping an eye on in Chesapeake Bay, and now we know that the SPAT tool that we've been using for many toxins might not be good for those toxins in particular, we're trying to explore another avenue. All right, so Josh, who you got to meet this morning, all of his work up till now has been done with the typical SPAT, the resin-based HP20 resin. But right now, as part of his dissertation, he's moving towards trialing a new passive sampler. It's made out of a patented mixed phase polymer, and that is already being shown to have better retention of those polar toxins, so a stronger affinity, and they can hold on to those microcystins, domoic acid, for like 28 days or more. So this has a lot of promise. SPATs, I think, are fantastic for those lipophilic toxins, so the okadaic acids. It's a tight binder of pectenotoxins, brevetoxins, azasparacids, goniodomins. But when it comes to microcystins and domoic acid, this might be a good alternative. So Josh is going to be working with that. We submitted a MURHAB, which would allow us to hopefully have collaborations with VDH and MDE so that we could field trial this new passive sampler. And if we're lucky, that would also mean collaborations with California and Florida. So we would get some of that brevetoxin data, because I think we all have questions on, is brevetoxin something we should be worrying about in the Bay? And if it becomes an issue, how are we going to track that? So this passive sampler will be tested with that also, since there would be a Florida collaborator. And then Josh is going to move forward with testing the stability of these toxins in water and their stability, the toxin stability on the new passive sampler. So is temperature degrading it, or hydrolysis, or is it nice and stable once it binds? Last slide. So for the third thing that we're moving forward continually in 2023 is the toxicity of phytoplankton biochemicals. So that's what you've been hearing us talk about. So the toxins, the ones that we currently are working on, which would be Nour, would be pectenotoxins, iazotoxins, and asparacids. But we're also bringing in new bioactive compounds, which would be the polyunsaturated aldehydes, oxylipins, and all of these are oxidized products of PUFAs, or polyunsaturated fatty acids. So it's these chemicals beyond just the toxins that might be having an effect. And currently we're looking in oyster larvae, and then Josh is moving us into shrimp. So we'll let you know how that goes. And then Mart is really helping us oversee a hatchery project that was just funded, which is looking at, it has more of a focus on the bioactive compounds, so those oxidized products, and possible effects in the shellfish hatchery failures with our collaborators that I listed there. Okay, so my email's on the bottom if anyone has any questions or wants some images or anything, and I couldn't help but end with a picture to give credit to everyone who does the work. So thank you for your time.
Thank you, Juliet. And I think we have time for any questions for Juliet.
Wade has a question online. He asked for SPATs, are there any attempts to use other resin types other than HP?
HP20? Yeah, was that the whole question? I should just say. So Wade, hi. Not in our lab. However, my understanding is that, so the MURHAB that we've submitted is with Reece Kudela, and he is relying on, he would be leveraging a MURHAB that he currently has funded, which is allowing him to test many different types of of resins, and even I don't want to speak for him, but I think he's also testing different types of enclosures for the resins or other materials. And so this is us kind of going on a whole new trajectory by using kind of more of like a piece of plastic, essentially a polymer plastic that you would submerge as opposed to the resin beads. So yes, that's happening. That is not us though.
Are there any more questions in here?
Thanks, everyone.
Thank you. Next here, we have Leah Anne Gibala-Smith, the supervisor from Egerton Phytoplankton Analysis Laboratory. She's going to be presenting on expanded capacity of biotoxin analysis and imaging technology. This drag it over. Will that be shared with them?
No, go back. Sorry. We can be the strength of the other.
The drag it over.
Oh, there, yes, it has to be there.
Thank you. Thank you, Leah.
Thank you. Okay. So then everything's good.
Yeah, right.
But you can see that.
Oh, yeah. Folks. Okay. Hi, everybody. I'm back for part 2. And Margie, I'm going to invite you to jump in to add to anything I touch upon. Because Margie knows a lot more about some of what we have in the works, looking for additional projects and collaborations with others. And I'm just gonna real briefly tell you about a few things that we're in just the beginning planning stages. We've just started doing, and some goals to kind of dial this stuff in the upcoming year. But building capacity for biotoxins analysis in the lab, and also using imaging technologies in ways to help us do our work and support our state partners. One of the things that we've been doing for a number of years is assisting NOAA with their building their hyperspectral satellite products and doing validation of some of their images. Margie just had them come to campus, and they did a visit a couple weeks ago.
No, they were here at VIMS.
They were here at VIMS. They were at VIMS, but they also gave a talk.
Or Shellfish
yeah,
gave it
yeah.
for us.
And so we've been working with her with doing some validation. A lot of the data that we generate is specifically is taxes specific to dinoflagellates. And a lot of our shellfish samples we don't count the other groups. And we are expanding some of our support for these products in using some of our imaging technologies. So even though we're doing our microscope work, looking for the HAB species, looking for the dinoflagellate, we're also running those samples through our ICP or our FlowCam or our Plantoscope, and we're building libraries that include other species. And so eventually what we hope to do is get cell count or biomass estimates of these other groups that have different types of pigments. And so we did a test of, I think this was maybe 2 or 3 years ago. Catherine and I did a full taxonomy on a large flume on the Rappahannock, and they found that very useful because with in addition to cyanos, in addition to dinoflagellates, so that's something we're trying to work on a little bit. Margie and Catherine, in support of Aaliyah, some of her culturing experiments, developed a new biotoxin safety protocol. And so we're hoping that gives us, expands our ability to do some additional toxins work in the future. We've been building our capability for growing toxin producing cyanobacteria in culture. That was Aaliyah's master's project, and she's looking to expand a lot of that for her doctoral work. And Margie's been talking to some people about that as well. And building those image libraries, like I spoke to you about previously, we're running our shellfish samples on the microscope as normal, saving them, storing those images, and we're hoping to use that to build a large library for coastal and tidal water in Virginia. And we're doing that in collaboration with some other states, with Maryland and Savannah. Yeah, and Savannah, yeah, and others. And Margie and Savannah both have talked about Pectenotoxins, and so we're exploring possibilities to incorporate all these different types of imaging technologies in monitoring efforts, as well as detection of novel potential HABs. And we're working with Kim and BDH for doing some cell counts to correlate with their qPCR and primers. And that's about it. Short and sweet.
And for those of you who don't know what a Pectoscope is, it's a low-cost flow-through imaging system that you can take in the field. They cost about $2,000 to build the first one and $1,000 after that. And so Kim's building 3. We have 3 working ones now, but they're nice, cheap, low cost.
Yeah. They take really good pictures too.
Yeah. And they just come out with a new version of on the Pectoscope website. And buy it, so we're gonna get parts of the new one. So I think it's mostly better camera.
Are those automated too, like you're getting, or is it
It's like something about the size of a portable hard drive. So it's like that square and about 2 inches thick. And then it's just, and it looks really low tech. You have a syringe on the top that you put a sample in, and then there's a small peristyle pump inside that pumps the water through and takes pictures. It's cameras. Yeah.
How quick is it?
Miniaturized, what's that?
How quick is it?
Pretty quick. Yeah, I mean, I think it takes them about, depending on how much volume, of course, they put through, but I think it takes them around 5 or 10 minutes to run a sample of about 5 mils.
You don't have to take a Todd, so. We'll get it.
But the House proposal we put in was we would build like 5 or so new ones a year and provide users with tablets and get them out of the field. It would help expand, the monitoring network.
And you're looking at putting them on boats too.
On boats?
Yeah.
Yeah, like the aquarium or with hatcheries or growers,
Yeah.
mediation, DEQ people.
People that are on the water.
Yeah, and our trusted users, and we provide them a tablet and they could have it upload to EcoTaxer straight away. And once we develop the image classifying, then we could have,
Then
quantify
can check
quickly, or at least quantifying quickly species of interest.
for the ones where you're running the VDH samples through, like those are all Google samples?
Those are, yeah, so I guess
That's
I
the
could have
works
said,
for-
yeah, we have to build 2 libraries. One library with fixed sample and one library for live or unfixed sample.
Yeah.
So there would be 2 image libraries.
Yeah, we're trying purposely to build 2 because most of the IFCDs are unpreserved
Right.
samples that people don't want to stick the preserved samples in them.
Yeah.
So the flow cam, we can go back and forth
Right.
between preserved and unpreserved. Same with the Pectoscope.
And I think it's useful to have, yeah.
Yeah, so we should find forces with at least, getting quick identification now for the HAB species of interest, at least the low hanging fruit.
The regular blooms.
So how many images does that plantoscope take of the 5 mils? Can it,
Everything in the water.
so thousands of images?
It's
In-
depending
in
on, I
5
think, the
minutes?
setting.
Settings.
In 5
Well,
minutes?
But they're pretty, the quality images are, they're not.
Gets fouled up all the time.
Yeah, the quality is really surprisingly good. What it doesn't do that somebody, the high price internet does, it doesn't do like the image analysis size, and different properties like that.
And then the plantoscope is more like a low cost.
Yeah.
Well, I'm sure. And they're still pretty, cheap, but not the, it's usually something else.
It's good. Yeah, without reading glasses, they would.
As ice.
Oh, right, yeah. Yeah, we can't get quite as small or like the screen with the peak of something really small.
The pictures that they did in the freshwater look really good on plantoscope.
Yeah.
The ponds.
Yeah.
I'll go ahead and do his introduction while we get him shared over, but up next we have Paul Zimba, who's a research faculty at VCU's Rice River Center with the Department of Biology at VCU. Paul graduated with a Master of Science from H.E. Marshall's lab at ODU, worked in the Horns Point Lab, Tom Malone, Richard Ripken, and Mike Kemp, and completed a PhD with Mike Sullivan on salt marsh algal community structure, algalotrophy, photoheterotrophy, and C3, C4 carbon fixation at Mississippi State University. He decided to work on Lake Okeechobee ecosystem study at the University of Florida in 1989, as well as on seagrass and the fifth bite sites. Then in 1996, with the USDA ARS on toxins / off flavors in aquaculture systems. 2009, he moved over to Texas A&M Corpus Christi, serving as the director of the Center of Coastal Studies and a professor in biology. He has mentored 7 PhD students and 12 master's students during his career. He retired in 2021 and moved back to Virginia to deal with mental health issues. And then in July of 2022, he was hired as an adjunct faculty member back at the Rice River Center at VCU. Then was published over 130 papers on stress photophysiology, photosensing algal toxins, cyanobacterial taxonomy, and remediation of harmful algal blooms. This includes 3 nudicides toxins, including bucoliphysine, a potent neurotoxin showing anti-cancer sensitivity. Then,
Okay, Paul? Yeah. If you want to put it on presentation view, then we can see you. Okay, Paul?
he moved the screen.
It's him.
It's moving.
That's him and not me.
It's not.
Hey, Paul, we can't hear you yet, so.
Something.
Hello,
Hello, now we can hear you. Can you put
so
your,
sorry for the. Go ahead.
can you put your PowerPoint on a presentation?
I
You-
believe it is.
Not-
I am screen sharing right now.
Yes, but we see the slides show. We don't see the presentation view. Maybe you're sharing a different screen. There you
Better?
go. Yeah, that's better.
It is.
I will move it. I can do that. Okay,
Thanks.
you're good.
Having your mouse hand incapacitated makes for a little bit of a problem. I apologize. So 37 years ago, I was a resident here in Virginia and moved down around the Gulf of Mexico for much of my career. And my work has covered quite a diverse set of areas in the last couple of years. This gives you an idea of some of the topic areas. I've maintained a very active role with regard to HABs, but also quite a bit of work on remote sensing approaches, as well as just the physiology of algae generally. I would, as I indicated before, I decided to go back to work. Rice Rivers Center was conveniently located just down the street from where I live, and I've been able to maintain a number of relationships over the years, which has allowed me to continue work on ultrasound methods of control of harmful algal blooms generally, as well as toxin detection and community structure. You may not be familiar with ultrasound use. We've developed some indoor systems that we can grow 150-liter volumes of single species of algae and expose them to the same equipment that would be used in the field, and be able to look at how ultrasound is killing the cells. Water IQ is a company that has come up with some significant improvements in the design of the ultrasound systems, and we're working with that company to look at individual species for the damage that is occurring from ultrasound exposure. And we're doing that largely by looking at TEM ultrastructure, as well as photophysiology using a phytopam to look for early stages of effect on the photosynthetic potential of the cells. In collaboration with University of Florida, Dale Laughinghouse, we've got a full proposal to prepare for the test bed program being run out of Moat for this equipment, and so we'll be busy to meet the April deadline. In October, I became aware of the HAB issues in the Shenandoah, went up and sampled, and the river keepers up there were instrumental in keeping me from drowning. We collected a series of samples that included several types, basically the benthic mats from the area right around the Strasburg town park or city park location, 3 miles downstream of there. This shows the extent, and you've seen various iterations of these maps. But by microscopy, we were able to see that Spirogyra was the dominant green throughout the entire reach, and I collected basically the benthic cyanomats and also removed the epiphytes from the Spirogyra and analyzed those as well. We were able to, with this, then analyze samples for toxin production, and also using MySeq analysis, look at the community structure. This isn't fitting for some reason on my screen, but the first sample on the left is the benthic mat sample. The sample next to it is another benthic mat sample collected a few hundred yards downstream of the initial location benthic mat again. And the third sample are the cyanobacteria epiphytes associated with the Spirogyra that was collected within a meter of that first sample. And so we have begun the analysis of this data, and I do want to keep pursuing different databases that are available to get a little bit further down with regard to the taxonomy. But it's pretty obvious that the community is quite a bit different at the time of sampling, with Microcyra woolii being a dominant, and that had not been recognized previously. Microcoleus tonoplastes was something that I've been able to acquire from enrichment culturing, and it does not show any toxin production. So I'm hoping that we can continue pulling out single species of this assemblage and identify the source of the toxins being produced. So the MSMS analysis that was done was repeated for 2 samples at 2 different labs. We had a very high hit with regard to homoanatoxin being present at greater than 1 microgram per gram wet weight in the benthic mat samples. The epiphytes showed no toxins. The only other identified bioactive metabolite from the cyanobacteria was anabenopeptin in very high concentrations as well in this one sampling. I was able to talk with the USGS and with the Potomac River Basin about how to pursue the Shenandoah River research going on, and I've made a couple suggestions that I've listed here. The use of a hyperspectral system to map at high resolution, around 1 to 2 centimeter resolution, would be a great way to follow successional patterns in reaches of the river. We figure we can do, or it would be possible to do about 600-yard stretches of the river at that resolution, and with ground truthing, produce maps that would track the seasonality of the bloom formation. And secondly, it was my suggestion that MSMS be used to look for more than the 5 sanotoxins the EPA has listed. And in particular, we've been finding more and more evidence that nodularans are widespread throughout the United States. And with the current ELISA approach, it limits the ability to separate those. But there would be a tiered approach for analysis, and if there were hits, then the second level would be to use MSMS on samples. So an ELISA first, and then MSMS. I also hope that we could begin to identify the forms of or the species that were actually producing the toxins, and I that's again my suggestions. Doesn't mean that many people listen, but the SPATT sampling is a great way to try to normalize between the single sample sort of grabs, and I think using them as a volume integrated approach is a great way to try to get a handle on the production of toxins that occurs basically, which could be missed with a single point sampling. And so those suggestions were made, and there's a meeting, I think, scheduled next week that hopefully will help resolve what is the final decision of what will be done in both Lake Anna and in the Susquehanna River. And so I hope to have some better pictures next time. I thank Wade, Dr. Aishu Huang at the FDA for doing a backup analysis of the anatoxin, and also I talked with Lee Pinnell, who helped with some of the genomic work, and I think that that's a great addition to just the microscopy, given the difficulty with cyanobacterial benthic taxonomy to use as another means of looking at larger volumes and coming up with dominance within the benthic community. Thank you.
Do we have any questions for Paul?
I'm not familiar with the abbreviation MS.
I'm sorry?
Were you able to hear that question? There was a question about what the abbreviation MS.
That's
That's
I'm sorry.
So what do you have, how does that work?
How does the mass spec work?
No, what's the meaning of mass spec? So you run through mass spec and you run through mass spec again? Or what does that mean?
Excuse me, it's a quadrupole mass spec, and so a triple quadrupole. And so basically you run your samples through, get a parent ion mass, and then you fragment and you look for the fragmentation products that are typical of that compound, and the ratio basically of those gives you a confirmatory level of knowledge of the presence of that material.
So you feel that's more highly resolves the chemicals better? People in the room are saying yeah.
the gold standard.
Okay.
I'm not.
Yeah. Thank you.
You can sometimes distinguish different isomers.
Okay. Yeah, I noticed you did it. That's American names up there.
Paul, I'm going to read you Shelley's question so you can answer out loud instead of having to type it. She said, we are looking at the use of drones for benthic planktothrix in the Potomac. It was complicated given that planktothrix was growing in a community with Lingbia and macroalgae. I'm not sure you could tease out the optical signature. That also required clear water to reach the bottom. Thoughts, given your suggestion for overflight data? I'm not sure how deep the bloom in the Shenandoah occurs and how turbid.
So you're right in one sense that separating out species or even genera of cyanobacteria will probably be a little bit difficult, if not impossible, but it will be possible to map the occurrence of cyanobacteria versus green algae, let's say, or diatoms based upon their accessory pigments and their chlorophyll A to B ratio. The hyperspectral system we have is a 6 nanometer resolution, which will be great for being able to separate out those groups.
Can you ask Shelley where she's from in the Potomac for that work and stuff? Hey,
I'm
Shelley.
sorry?
Sorry, I was asking the full Zoom meeting.
Ellie, where are you? Did you hear that question?
I couldn't hear the question. What was it?
Oh, where are you in the Potomac?
Oh, so it was off Brunswick, Maryland.
Right.
We were working with DNR, and I went out with a handheld radiometer, and it was really hard to tell much of anything.
Was that Kevin's work?
Yeah, he was involved. Yes.
the Shenandoah is likely much more, much less turbid, I would think, than the Potomac. But I'm just speculating here. You know, from what I've seen, the amount of suspended solids in the Shenandoah, except under, you know, a flood period, is not going to be a confounding influence.
I can just speak for Brunswick as well. That particular stretch of river is, I guess, deeper and more complicated than where some of the blooms are happening in the Shenandoah. So the Shenandoah just being shallower, less features to I guess address the surface. This is another place to give that methodology a try.
That makes sense. Thanks.
Thank you.
All right, I don't know if there's any more questions for our previous speakers. We're good on time. I know it's also a Friday.
All right. Well, thank you, everybody, thank you to all the speakers.
Thank you for hosting.
You can drop your name tags off back at the desk out front, or just lay them on a desk here when you find one too.
No, take our name.
You can take it, yeah, you can take the paper. You can take the paper. By the way, the beautiful work here, that's thanks to Savannah. So thank you very much. Thank you, Margaret, for all the organizing. That's great. Any last minute questions? Any last minutes for Todd, Margaret?
That's where.
We will have the recording available. Assuming Margaret's going to send out all the information. We'll stop the recording and save it. Compile it and send it ready for you. It will have a lot of links. Please get over that.
Do we need to, you have the presentations up there, or do we need to get the presentation?
Some of the remote people will need to get the presentations to us unless Margaret already has them. I have the presentations for the speakers who are here in the room. We'll convert them to PDF. Some of them are still PowerPoint.
Like I said, Drew is still with us out there. Juliet, of course, she already knows she's gonna get me hers, and Paul, if you wouldn't mind emailing me PDF versions of your PowerPoint, I'd appreciate it. Thank you.
Sure.
Any other housekeeping? Please feel free to take snacks, cookies, fruit, whatever you need for the road. Okay. Great, thank you.
Have a good day.
That's nice.
Volunteer.